1 2 1 2 1 2 1 2. Mic check 1 2 1 2 1 2. Mic check 1 2 1 2 1 2. Mic check 1 2. Mic check 1 2 1 2 1 2. Mic check 1 2. Mic check 1 2 1 2. Welcome. Mic check 1 2. Mic check 1 2. 1 2 1 2. Mic check 1 2. Mic check 1 2. Mic check 1 2 1 2. Mic check 1 2 1 2. Mic check 1 2 1 2. Check 1 check 1 check 1. Mic 1 mic 1. Mic check 2 2. Mic check 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 2. Mic check. Mic check. Mic check. Mic check 1 2. Mic check 1 2. Mic 2. Mic 1. Testing, testing. Mic 1. Mic check. Mic check 2. Mic check 2. Mic check 1 2 1 2 1 2. Mic check 1 2. Try 2. Yeah, 2.
Okay, wait, now you can hear me.
Check 2. Yeah, 2. Okay. Check 2. Mic check 2. Mic 1 2.
Yeah. Once you've gotten to this.
Mic 2.
So is it too late to get a projector?
Hello?
Not too late. Not too late. If we can get a projector, it's not too late. In-house stage, I'm not sure.
Hello? Hello? Yeah. Hello? 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 Hello?
Am I am right? Yamo Yamberaye, Abeya, Efrianara. Asam Paetia, Obayansa Fusu, Bunia, one can asam. Say, Mafu to some Kakrewa Supeme. Yo, Kofi, Amaya woman, Amani Amamana, Yeni baby, Watata, Jayawaren, Wonsum, one in pa, as I did power of you. Yamo Yamberaye, Abeya, Amaya woman, Amaya Mamana. Asam Paetia, Obayansa Fusu, Bunia, one can asam. Say, Mafu to some Kakrewa Supeme. Yamo Yamberaye, Ebeya, Efri Yanara. Asampaitia, Obayan Sapusu, Bunia, one can asam. Say, Mafu to some Kakre was Supreme. Yo, Kofi, Amaya woman, Amani Amana, Yeni baby, Yako, Wachata, Jayawaren, Wonsum, Waninipa, Asadipa, Wofio, Yamo Yamberaye, Abeya, Amaya Umano, Amaya Mamana, Adiwaya Nora, Yamichata, Yamana Renya, Waso, Waninipa, Sawaya, Wanabe, Yamo Yamberaye, Abeya, Waso, Waninipa, Nasadipa, Ofio, Yamo Yamberaye, Abeya, Amabeya, Amabeyo, Asitebeya, oh, Amabeya, Yamo Yamberaye, Abeya, Obrani Warabo, Amabeya, Eminawa, Yamo Yamberaye, Abeya, Amabeya, Abraya was Abeya, Efrianaro, Ao, Amabeya, Efrianara, Yamo Yamberaye, Abeya, Yamoni, Amabeya, Efrianara, Abraya Boya, Yamo Yamberaye, Abeya, Amabeyo, Amabeya, Obrani Warabo, Yamo Yamberaye, Abeya, Can you bring the wafa? Timpawa Wayas Nawinya, Yamishira, Omani Paya, the Fua, and Yanum, Afanya Misroya, Jumana, Yami Besrayan, Obraya Boya, Yamo Yamberaye, Abeya, Efrianara, Amabeya, Efrianara, Ejanova Pofo, Asitebeya, Efrianara, Zemtrafwe, Amabeya, Efrianara, Adije, Yamo Yamberaye, Abeya, Yafa Ejibankawa, Amabeya, Efrianara, Yanaro, Yamo Yamberaye, Abeya, Efrianara, Ese Abrabeya, Efrianaro, Amijumaya, Asitebeya, Efrianaro, Amabeya, Efrianara, Yamo Yamberaye, Abeya, Yajobiyazo, Amabeya, Efrianaro, Yamo Yamberaye, Abeya, Saema Okarana Washawa, Amabeya, Efrianaro, Amabeya, Yamo Yamberaye, Abeya, Wasachimara, Wadongana, Amabeya, Efrianaro, Yamo Yamberaye, Abeya, Ajajabwe, Amabeya, Efrianaro, Yamo Yamberaye, Abeya, Efrianaro. Asampaitia, Obayan Sapusu, Bunia, one can asam. Say, Mafu to some Kakre was Supreme. Sampaetia Obayansa Fusu Obunia one kana sam Zemafu to some kakre wa sukume Yo kofi Amaya woman Amani ya memana Yeni baby ako Wachaza Jaya wareni Wonsom Wanini pa Asade pa Wofioye Yamo Yambera
Hello. Good morning, ladies and gentlemen. On behalf of Management of Ghana Atomic Energy Commission and the Biotechnology and Nuclear Agriculture Institute, I welcome you all to GAEC for this Cassava Week celebration, and then also as part of the GAEC 60th anniversary and then 30th anniversary of Binary. You're welcome. As you bear with me, the world has an owner, so we can't gather here without his permission. By so doing, I will invite Reverend Dr. Delali Koto to do us the honor by opening the program with a word of prayer. Shall we welcome him?
Good morning to you all. Please, can we be upstanding for a word of prayer? Let's bow our heads. God, our Heavenly Father, we thank you for the gift of life. We thank you for seeing us through the night which has passed and for giving us a new day. We give you all the praise and all the glory. Father, we rejoice over Binary and its achievements for the past 30 years. And today we are gathered here for the Cassava Week celebration, which seeks to promote the cultivation and utilization of cassava in Ghana. Father, we invoke your Holy Spirit in this place. We pray that your Holy Spirit will take absolute control of everything that we are going to do here and all the programs that has been planned. Father, we come against and terminate every agenda of the evil one. And we pray that the program outline and everything that will be done here will be successful. We continue to pray for the leadership and staff of the commission. We pray that you continue to preserve and bless our lives. In the mighty name of Jesus. We pray that may we continue to strive and work harder to better our lives and also to fulfill our mandate. These and many others we pray in the name of our Lord Jesus. And everyone shall say amen.
Thank you, Doc, for that wonderful prayer. Ladies and gentlemen, I usually call him the Odikro of Ghana Atomic Energy Commission.
Limited, Mr. Evans Ayim. Limited, Mr. Evans Ayim. Directors of Institutes of GAIC, former Director of Crop for Development and Deployment of Sustainable Agricultural Solutions for improving the cassava value chain. These efforts align with the government of Ghana's efforts to equity in cassava systems. We also have here partners from the industry, farmers.
Him out. A consultant for JSA, Farms, a subsidiary of the Jospon Group of Companies. He currently...
Good morning. Thank you. Thank you very much. Mr. Chairman, Director General, Ghana Atomic Energy, Peasant Farmers Association of Ghana. Operators for this Cassava Week celebration, Innovation Village, per soybean Gary blend, known as Pro Gary. All these efforts contribute towards ensuring food availability, accessibility, utilization, and systems all across the country. Because cassava just needs to sprout all across the country. Because cassava just needs to sprout. There's no rain, it will hang in there. As soon as it gets rain, we are still doing it the way we did it in the 1920s and 30s. Sin region processing starch, among others. Many of these factories, however, are not running at full capacity due to difficulty in obtaining raw materials, that is cassava, for production. Relevant on the field in a manner that benefits the whole nation. Mr. Chairman, we do not have to lose sight of our climate close Ghana. Other numerous challenges faced by the farmers in the cultivation of cassava include poor soils, materials elsewhere, and bring in about 10 million cassava stems for every 10,000 hectares to be planted. 20-30 seasons, and it's coming from one parent stock. Because we just harvest, cut it, and replant. Harvest, cut it, and replant. Harvest, cut it, and replant. So we are just spreading from agricultural institutions and our extension officers. It is important, however, that some people with experience in cassava research are involved to train local farmers on conducting simple cassava. We should be deliberate about how we farm it. So we bring the knowledge from the laboratory, take it on the field, production and yields. So someone will farm without the yield in mind. And when you go on the field, it is very weedy. And at the end of the day, he expects to have harvest. You cannot planted in a sequence in lines with a certain planting population, applied fertilizer, and expecting a certain yield. And you just went and slashed the field, put the sticks there, and you won't harvest. So, and getting very high yields. And one factory I visited produces 300 tons of gari a day and export everything to the US. So researchers must not be reinventing the wheels, but work together to find solutions to our everyday problems. And we must work for local solutions to our local problems. I've seen in other places that they don't complicate farming. They use simple the Cassava Week celebrations organized by Binary Gaek and his collaborators to promote the adoption, cultivation, utilization.
Ladies and gentlemen, we go back to IAEA to join Professor Razak. Bidro Kakraba. Bibidro Kakraba. What's up? Yeah.
We also have Joseph Edu Jemfi, IAEA. Ladies and gentlemen, it is said that behind every successful man is a woman. We have the wife of our Director General here, Mrs. Some songs. Whoa, whoa. When I say big up, big up, big up, yeah? You know it already. Okay, so let's do this one. Yo, big up, big bunchy, bunchy. Bunchy, bunchy. Bunchy, bunchy. Big up the cassava farmer. Big up, big up, big up, big up, big up. Said Rade, said Rade, said Rade. If you love me, said Rade, said Rade, said Rade. All right, selector, if you're with me, let's do this one. Okay, selector, let it go higher. Can you pitch it higher? Oh, day, health is wealth. Hey, hey. Organic to the world. Big up cassava farmer. Bigger. Ladies and gentlemen, we also have in our midst Theodosia Adom. And then a rep for the director of Ghana Irrigation.
I must say that the Cassava Week celebration is highly recommendable, and it's very good. In my personal opinion, I think we must also take note that cassava, to me, is one of the food produced almost organically. So it is near organic food. Because, Director General, I'm very happy that this program has been initiated, and I pray that it will continue to help transform cassava.
Thank you so much, Ray. Before I do the vote of thanks, I would like to acknowledge our partners in this program. And then all at the high table, on behalf of Binari and the Cassava Celebration Committee, we appreciate your coming here and the knowledge you've shared.
Committee, and then she has come all the way from Kumasi to be part of this program. We have a wonderful personality in the name of Mr. George Pra. Please, are you here? Good. Give him a round of applause. He has come to be part of this panel discussion. So, be part of this panel discussion. So,
Thank you. Auntie Angie, please, you have to join, or I'll say it in the mic. Please, you join there. There's a cassava break. What are
Asampaetia Obayansa fuso wo munuye wonka na asam Te mafutu sam kakre wa asupeme Yo kofi amani ya woman Amani ya memana Geni baby ako Wachata jaya wareni Wonsom wanini pa Asati pa wofioye Yamo yambera ye Ebe ya Yena Amae ya umano Amae ya memana Ati waye nora Yamichata Yamana renya Wanso wanyinyi pa Sawa ya wanabe Yamo yambera ye Ebe ya Yena Wanso sabu wanyinyi pa Nasa pa ofioye Yamo yambera ye Ebe ya Yena Amabe ya Yena Amabe yo Asite be ya Yena Oh Amabe ya Yena Yena Yamo yambera ye Ebe ya Obra you arabo Amabe ya Yena Emina Yamo yambera ye Ebe ya Yena Ese Amabe ya Yena Abraya we sabeya Efriana ro Ao Amabeya Efriana Yamo yambera Abeya Yamoni Amabeya Efriana Abraya we Yamo yambera Abeya Amabeyo Amabeya Efriana Abani warabo Yamo yambera Abeya Efriana Sikanye bredo wafa Timpawa weyes na winya Yamishira Omanyi payo zefua Enyano Yafanya misronya Jumana nyami besrayen Obraya boya Yamo yambera Abeya Efriana Amabeya Efriana Ejano mapofo Asitrebeya Efriana Zemtrabwe Amabeya Efriana Adije Yamo yambera Abeya Yafa enyebenkawa Amabeya Efriana Yeraro Yamo yambera Abeya Efriana Ese Abrabeya Efriana Amijuma Asitrebeya Efriana Amabeya Efriana Yamo yambera Abeya Yajobiya yazo Amabeya Efriana Efriana Yamo yambera Abeya Saheba ukarana washawa Amabeya Efriana Amabeya Yamo yambera Abeya Wasachima wadonkela wasa Amabeya Efriana Efriana Yamo yambera Abeya Ajaja we Amabeya Efriana Yamo yambera Abeya Efriana Asampaetia Obayansa fuso wo munuye wonka na asam Te mafutu sam kakre wa asupeme Yo Sampaetia Obayansa Fuso Munia one kana sam Zema to some kakre wa supreme Yo kofi Amani ya woman Amani ya memana Geni baby ako Wacha ya wareni Bonsom wanini pa Asa dipa wofyo Yamo yambera Mama Sampaetia Obayansa Fusu Obunia one kana sam Zema to some kakre wa supreme Yo kofi amaya woman amani ya mama Yeni baby ako wacha ya wareni Wonsom wanini pa asate pa wofio Yamo yambera Sampaetia Obayansa Fusu Obunia one kana sam Zema to some kakre wa supreme Yo Kofi Amaya woman Amani ya memana Geni baby ako Wacha ya wareni Ponsom wanini pa Asa tipa wofyo Yamo yambera ebe ya ifuyena Amaya umano Amaya memana Ati waya nora Nyamichata Nyamana renya Waso waninyipa Swawaya wanabe Yamo yambera ebe ya ifuyena Waso waninyipa Satepa ofyo yo Kamo yambira ebe ya ifiyana Ama be ya ifiyana Ama be yo Asite be ya ifiyana Oh Ama be ya ifiyana Yana Yamo yambira ebe ya Obrani warabo Mama Beya, Efriana, Eminawa, Yamo Yambera, Ebeya, Efriana, Mama Beya, Efriana, Mafraya was Abeya, Efriana, oh, Mama Beya, Efriana, Yamo Yambera, Ebeya. Can you read the wafa? Timpower warriors now winya. Yamishira, Mamani Paya, the Foa, and Yanum. Afanya Misroya, Jumana, Yami Besrayan, O'Brien, where Yamoyamura, Abeya, Efriana, Amabeya, Efriana, Ejanova Pofo, Asitrebeya, Efriana, Amabeya, Efriana, Adije, Yamoyamura, Abeya, Yafa, and Yebunkawa, Amabeya, Efriana.
So, good morning once again, and welcome to the first of two plenary sessions this morning and later on in the afternoon. For these sessions, we have distinguished presenters take us through some pertinent topics on Cassava, and we'll have an opportunity to engage with them and get a better understanding of what their work has been and what Cassava has contributed to their working lives. For the first session, we have two presenters. One presenter is currently here with us, and the second presenter is joining us from Abidjan in La Côte d'Ivoire. So I would want to give him the opportunity to present first, and then our second presenter would go. Our first presenter is Professor Emmanuel Yauvi Bobobi. He's an associate professor of agricultural machinery engineering in the Department of Agricultural and Biosystems Engineering at the Kwame Nkrumah University of Science and Technology, KNUST, Kumasi. From 2013 to 2015, he was a specialist researcher in agricultural mechanization at the Agricultural Research Council's Institute for Agricultural Engineering in Pretoria, South Africa. Prof. Bobobi has consulted for a lot of organizations, including the AUDA NEPAD, AU NEPAD, IFPRI, FAO GIZ, World Bank, UNOPS, and ANADE of Côte d'Ivoire, and especially for the Ministry of Food and Agriculture in Ghana. He is currently the coordinator of the ARIPO KNUST MPhil program in intellectual property, and he's been in that role since 2018. He holds a PhD in agricultural machinery engineering from the Swedish University of Agricultural Sciences in Uppsala, an MSc in agricultural machinery engineering from Cranfield University in the United Kingdom, and a BSc in agricultural engineering from KNUST in Ghana. He has vast experience in agricultural and biosystems engineering, agricultural equipment development and mechanization, and he's invented the innovative tech mechanical cassava harvester. That harvester is currently in use in Ghana, South Africa, Nigeria, Jamaica, and La Côte d'Ivoire. And it's ideal for harvesting tropical root and tuber crops like cassava, yam, sweet potatoes, cocoyam, and Irish potato, for which he was shortlisted by the Royal Academy of Engineering, UK, among 12 finalists for the 2016 Africa Prize in Engineering Innovation. For that same invention, Prof. Bobobi received the first Engineering Excellence Award from the Ghana Institution of Engineers in December 2014, and the third prize from the Royal Academy of Engineering's Newton Fund for Leadership and Innovation Fellowship in March 2015. He also developed the tech granite cracker winnower that cracks and winnows granite in a single operation. He's authored over 50 scientific and technical publications in agricultural engineering and has traveled extensively to several countries to present scientific papers. He is currently a member of several scientific and professional bodies. He's a council member for the Pan-African Society for Agricultural Engineering. He's a fellow of the South African Institution of Agricultural Engineers and a senior member of the Ghana Institution of Engineers, and a founding member and past executive of the West African Society of Agricultural Engineers. Prof. Bobobi's goal is to impact his in-depth knowledge and rich experiences in the youth of his generation in Ghana, Africa, and beyond, and he works tirelessly to bring great change in the agricultural sector in Ghana and in Africa and the world at large. He has learned to give back to society through his philanthropic and charitable donations to his community, students, and his generation. And he's married with children. Probably didn't add this, but he was the vice presidential candidate for the Convention People's Party in the 2020 elections. So he's not just an academic, but he's also politically minded. Okay. So Prof. Obi will be taking us through the emerging technologies for cassava production mechanization. Prof, your audience.
Hello, can you hear me? Yes, we can hear you. Hello?
Yes, we can, Prof.
Okay. Thank you, Prof, for that presentation of me and then my political decision. I had a slide on that, but I have to remove it last week. Anyway, good afternoon to you all, and then I'm standing on the previous protocols that were presented by others. So I'll go straight to the presentation because of limitation of time. So let me play my presentation now. Please, can you see it? Please, can you see my presentation? Hello?
No, we can't see it.
Hello?
We can't see it, Prof.
You can't see it?
Yes.
Oh. What is the problem? Because trying to. Still you can't see.
So Prof, please share your screen. Please share your screen.
I'm sharing it, but. Oh, goodness. Can you see it? Can you see?
Yes.
Please, you can see now. Can you see, Prof?
Yes, Prof.
Okay, thank you. In fact, I have two topics, but I've made a last minute change because of the coordinator. And they are all the same. That is potential of cassava as an industrial crop. My name is there, and then my emails are also there, and I have a website also, which is the techcassavaharvester.com, which you can access on YouTube and other places. Yeah, I think I'll go straight and then list some few challenges to large-scale cassava commercial production in challenge to large-scale commercial agriculture in Ghana and Africa, including cassava production. First of all, the smallholder agriculture dominates on the continent. When the use of back-breaking hand tools are predominant, agriculture is a way of life not treated as a business, and agricultural labor is aging in Africa, and the youth are not interested to invest their talent and energy into agriculture. This is a typical farm power typology and hunger map. That is, on the left, you can see the farm power. Countries that use low farm power are actually countries that are prone to hunger. It means there's a strong correlation between the use of energy on the farm or farm power and food availability and the prevalence of hunger. And we are among the countries that use low energy on the farm. In Africa, it's only in South Africa, in Sub-Saharan Africa, that appears to have a resemblance of the first world country because of its high level adoption of mechanized agriculture. So, and this is true that mechanization is one of the ways to go in the new fourth industrial revolution. So the objective of this presentation is to introduce, demonstrate, and popularize mechanized cassava production and harvesting technology to unlock the huge potential of cassava as a nutritious and multi-billion industrial crop in Africa. So I'm talking as a typical African academic and my journey, some of my experiences. So this slide is telling some challenges that we face, and then some of the innovations that I have done or participated in doing. So we have the research area, that is the focus of the research, the challenges we are trying to provide solution to, the innovation that has come out of it, and then the present status of such innovation. And here you can see that we are talking about cassava harvesting. The challenge is that this cassava harvesting is tedious and embroidery. And the innovation is a mechanical cassava harvester with a durable blade. The status is that it has been patented and commercialized. Then, for the other one is to do with a rigid development, and this is also to support cassava production in line with climate-smart agriculture. Then the next one is cassava peeling and slicing for smallholder farmers. It is laborious and time-consuming. So we have developed a prototype, and then it is at the testing stage. I've really done some work in granule cracking and winnowing, develop a motorized granule cracker, and then even now I'm working on tiger nut harvesting, mechanical tiger nut harvesting. This prototype will be ready this year, and then to be tested in the field. And then we'll modify and then maybe release it later on. So this slide is telling us about the global agricultural innovation and mechanization progress. We have the starting here, we have the bending to work, like typical in Ghana. And this dates back to the time of Adam and Eve in the Bible. Then later on, we have the use of animals, like domestic animals, to provide motor power. Then in the 18th and 19th century, we have the steam engine or the external combustion engine. And that is the beginning of the first industrial revolution. Now, the dotted line is showing the drudgery. And the drudgery is actually the pain involving, the physical pain involved in doing any work. So if you are using mechanized technology, the drudgery level falls. And that is what we should aim at. Outside the slide, we have the second industrial revolution, which is the time of the car, internal combustion engine, and electricity generation, followed by the second industrial revolution, which is microprocessors, the development of computer. And now we are in the fourth industrial revolution, where we are using frontier technologies like robots, robotic technology, artificial intelligence, machine learning, Internet of Things, cloud computing, and all what we are all used to now. Even now the chat GPT happening, which I just started last year, November. So the question is, what contribution are we making as Africans to all these developments globally? So I would say that the plow, as an agricultural engineer, I would say that the plow kick-started civilization and ultimately made our modern economy possible. And for that one, for lack of time, let me just go to the benefits of cassava. Cassava, we all know, is an important crop nutritionally. It is food to over 1 billion people on the globe, according to Sustainable Goal. Goal 2. Cassava has high carbohydrate to be converted into several industrial products, including bioethanol and others. That satisfies Sustainable Goals 1, 2, and 9. That is poverty reduction, elimination of hunger, and then infrastructure, innovation, and entrepreneurship. Africa is a global leader in cassava production, but it is the least exporter of cassava products. Why is this so? And if you look at the global map, this is the global distribution of cassava. The dominant producers are in Africa. In fact, it is now about 60% is produced by Africa. And the Southeast Asia is about 30, and South America is about 10%. But when it comes to the revenue stream from cassava, Southeast Asia, especially Thailand, actually is the leading country that exports cassava products globally to China and then the West and other parts of the world. But Africa, we are not there at all. In fact, that is something for concern for all of us at this meeting. So these are the 10 leading producers of global producers of cassava. And Ghana happens to be the number 4. And you can see that we produce about over 22 million metric tons. And then that is not bad, considering our size. But all this production, the problem is, why are we not adding value? And then what are some of the benefits that we are missing? This will be discussed in some of the slides that will follow. So these are some of the industrial cassava products. On top of the range is bioethanol. And I think we have 2 factories in the country which are producing this. The one in Ho, that is Caltech. And then the one at Joapon, I mean, that is at Sanostone Bioethanol. And then the rest are producing other derivatives like starch and flour. And I think the Cassava Special Initiative that also President Kufo was actually the best thing that has happened to cassava in our country. That is when we all knew that cassava has got additional industrial potential, apart from just it being a raw material for food and other cassava products. So we have artificial rice, animal feed, paper industry, the food industry, confectionery, textile industry, monosodium glutamate sweeteners in the pharmaceutical industry as an adhesive, bioplastic, and even the cassava chips like this, Indomie, spaghetti, and the rest. And some time ago, Ghana, they were talking about plastic rice. I think, if nobody can boil a plastic, and most of those rice, maybe artificial rice, coming from cassava starch. And this is a study I've done in my research field to say that the starch content of cassava increases from MEF 7 after planting to about MEF 12. And then they start decreasing thereafter. The benefit of this slide is that we don't need to leave cassava on the farm for more than a year. So we can treat cassava as an annual crop now and get a huge potential from it. And the revenue streams, this is just a casual calculation I've done. The revenue streams from some cassava produce. So we take gari, high quality cassava flour, starch, and ethanol. And if we assume the yield is 30 tons per hectare, especially the lead cassava produced by our colleagues at the Crop Research Institute, CSIR, then we are just assuming that if we take 1 million hectares of cassava, we put 1 million hectares of cassava, and then we turn all that cassava into gari. From that 1 million hectares, we will get about $3.36 billion. If we convert it into high quality cassava flour, we'll get about 4+ billion. Starch also about 4+ billion. And if we take it into bioethanol, we'll get between 4 to 5 billion a year. And this compares with the area we put under cocoa. And the revenue from cocoa, the global revenue from cocoa is about $140 billion a year. But Ghana, as the second leading producer, we get only $1.3 billion from cocoa every year. And that is, you can hear that from the syndicated loan that we get. So it means we have allocated the most fertile part of our land to grow the crop cocoa as our main foreign exchange earner, but it doesn't contribute to our diet. But the one that contributes to our diet, we grow them on marginal lands. I think I listened to the remark by Professor Danso about why cassava is, you don't need to spend too much on cassava production. There's a crop we have neglected. So I think Binari, you are doing very well by creating this awareness. And if we all buy into it, cassava will become a multi-billion cash crop in this country and even on the continent. So the uses of modified starch are also the mentioned some already. Then what are some of the challenges to commercial cassava production in Africa? We have cassava is planted randomly. This traditional planting is not mechanized yet. And also manual harvesting is predominantly painful and stressful and full of drudgery. And until recently, there are no commercial mechanical cassava harvesters in Africa. So this is haphazard planting. There's no way you can mechanize, weeding and harvesting because the tractor cannot go into the field. So this habit must be changed to make cassava an industrial commercial crop on our country, on our continent. So the development of the mechanical cassava harvest has been so if you look at the production tax, harvesting is the one that takes the highest man hours or man days. As for land preparation, now we are using tractors, so there's no manual preparation per se. But harvesting, there's still manual harvesting, and when the ground is hard, it is a big problem. So Ghana is 53 hours, man days, and then Nigeria is about 62, Uganda is about 52, Ivory Coast about 44, and DR Congo is about 48. So harvesting is the highest, tax, energy consuming tax in the cassava production calendar. So something must be done. And this is how cassava is harvested manually. If the soil is soft, you can handle it like this on the top left. But if it is hard, two strong men must combine their efforts to uproot. Otherwise, you have to use the hoe and the cutlass to habit the soil. And this will not give you any high, productivity or efficiency per farmer. And these are some of the difficulties in it. And then look at this beautiful lady. Look at the way she's struggling to uproot one cassava, maybe for home consumption. Look at that. That is terrible. And this must be avoided at all costs. So my research area in all this is drudgery evaluation. Drudgery. And that is the physical pain involved in doing any manual work. And I'll talk about this later. So when you reduce drudgery, then the whole production process will be, user-friendly and interesting. And it will be attractive to the youth. This is a profile of two women in my own household who went to uproot cassava. They sat in a car, then later on they were uprooting just 10 stems. They rested, then I detached, and then they loaded into the car. Then after that, we were going home, they sat in the car. You can see how low their heartbeats were. And when we came home, they have to offload. This is hard. So the heart is one organ in our body which, and with clinical precision, predict the pain we suffer in any physical work. And this is one way I have been using, one parameter I've been using to actually estimate drudgery in agriculture. So the solution is that the tech, we have developed a mechanical cassava harvester, and this thing has been patented. It weighs about 280 kilograms. The harvester takes about it harvests 1 second to harvest a plant, compared with 5 to 10 minutes per plant manually. I produced over 50 prototypes, and 34 have been deployed in Ghana, South Africa, Nigeria, Cote d'Ivoire, and Jamaica. And the capacity exists to manufacture to satisfy continental demand. On the left is a patent, which is an international patent. Then on the right, these are some of the awards given. These have been mentioned already. So this is a short video of how the harvester works. For those of you who have not seen it before, it works best when the ground is hard. And it doesn't break the cassava. And then the advantage is that after harvesting, because this goes deeper than the plowing, the field is plowed. So you escape what is happening. Prof, am I still online?
Yes, you're still online. But your presentation is not showing.
Okay, I will wind up soon. So I think we have seen the mechanical harvesting. So this innovation we have developed when it is coming to actually remove all the drudgery involved in cassava production, especially harvesting all year round when the ground is hard. And this will satisfy both food security and industrial production. Some challenges here.
Yes, so Prof, your presentation is not showing. We see you, but your presentation is not showing.
So I'm now, has it come? No, wait. Is it on now? Can you see it? Prof, can you see it now?
No, Prof.
Okay, there's something. Let me just. Has it come? Has it come?
No, Prof.
Prof, has it come?
No, Prof, it's still not visible.
I don't. What is it? My. No, I think. Let me just work it out.
Yes, yes, it's so. So if you can put it in presentation mode. If you can put it in presentation mode.
I've done it now.
Okay.
So this is also another, sorry about the interruption, this is also another video of mechanical harvesting when the ground was very, very hard without, it was just about 1% moisture content. And this was able to do it with ease. This slide is a typical slide I picked from the work I did in South Africa, where the two top slides, the black and the blue, represent the heart rate of two women, Regina and Emily, when they were harvesting just 8 plants on the same day. The red profile is for the tractor operator, his heart rate. And you can see how low this heart rate profile is. The move one is the heart rate of the same tractor operator the year before when he was harrowing. This confirms that harvesting with the device, harvesting cassava with the device and then harrowing is a very easy task. And this tractor operator harvested close to 1 acre. Where this woman used the same 70 minutes to harvest 10 plants. So you can see the huge amplification in the adoption of the mechanical harvester. So this is a mathematical equation, governing the drudgery. So the P here, if the heart rate of the worker is high, as the denominator here, it will make this, let the right-hand side of the equation very small. And with that, it means the person must rest for a very long time. Which means that if the work is tedious, the person cannot work for long hours. That's about maximum 2 hours a day. And that affects the productivity. So if we can get the heart rate to be as close to, say, the energy consumption to be, say, 250, is the person may not need even to rest at all. It's just like working on our laptops or using our phones. Very, very easy. And that is where we have to aspire to take cassava production to all agriculture in Africa to make it smart and sexy. So this is when after harvesting, this is the way the cassava is exposed. You just pick it, and then the field is already plowed. The same field, the same example that you don't need to plow again. The damage is very low. Now this is showing that it can plow very deep, so you don't need to plow or to harrow before you reach again. So you escape the energy and then the clear way for planting, for plowing and harrowing. So this is also a conservation agricultural practice, and it is climate smart. I mean, technology. These are some examples of the benefit when you use the harvester, the plow, or the harrow. They are all in the same range. The good is that this harvester does not require any high power rated tractor to pull it. Yeah, because if you look at the one for the engine power required, I mean, the power it requires is even lower than that of the harrow and then the plow. So this has been made specifically for the African farmer in mind to use a tractor that is available in its environment, and it is something that must be adopted on the continent to lift many cassava farmers out of poverty. These are some media coverage on the technology when it was outlawed in 2011, both in the graphic and the Times. Additionally, the device can harvest even yam, but it must also be planted in rows or ridges. And that is good for the export market. Where you don't need those ceremonial ones that people outside can consume. It is also suitable. You can harvest potato or even a added potato. So it will come to mechanize and then remove the pain in all these roots and tubers. Now, if you look at the whole device, it has been rented. There's no spare part, there's no moving part. It is only the disc, the blade that you can replace. And this is something that I actually use my PhD thesis to investigate. So this can last for as long as we do, and I've made it in such a way that it is reversible. So if one side is worn, the other side will be used. So the metallurgy and the method of all these are part of my PhD work. So before this thing wears out like this, we know how many hectares it can do. So the future of cassava in Africa is that the future is we have to plant cassava on ridges, whether smallholder farmer or commercial farm. And once we do that, the farmer will very little to do on a day-to-day basis, and cassava will be available to satisfy the food security requirement and for industrial processing. So this is the planting on the ridge. The ridge is recommended, and it has got also climate smart advantages. Like if it rains heavily, the excess water will be evacuated through the furrows, and that is a benefit so that the crop is not lost, especially when it matures and then the roots will not actually withstand flooding for a day or two. And the spacing has been recommended
for now, to mechanize cassava harvesting, the spacing is slightly different from what our agronomists have been recommending, 1 meter by 1 meter. This time, it must be wide enough for the tractor to pass through. And then you will decrease the plant distance so that you get the same plant population density. And this is something that must be adopted, and you will not lose any yield. You may even get more, because when you reach, our colleagues from crop research have confirmed that the yield of cassava on ridges is higher than planting on the flat. And you can mechanize all activity, weeding and harvesting. This is a drone area picture of my field in the university. I planted all the 22 elite varieties released by crop research, some years back, and then I tested their compliance to mechanical harvesting and everything, and all of them are very good. There was one particular variety, like known as Guadipapa. That particular variety can yield to about, say, 70 to 80 tons per hectare. The only thing is that if canopy is not able to suppress weeds, so you may spend a little bit more in controlling weeds. Otherwise, it is the highest yielding cassava variety I've planted. So commercial farming is also they are adopting it. So not the problem, but the bulk of cassava is at the small holder farm. You need to actually these people by teaching them to adopt the. Okay, when I be talking about pineapple, and it is too narrow for cassava, because if you use that for cassava, harvesting will be a problem. So what I did was that I got one of my master students to be involved in it, and then we produced a ridger for double row ridger for cassava ridging, and then it is something that we are actually going to modify soon. So that is how it works, and this is from my students' project. So in fact, one commercial farmer is using it already. We are going to produce more. So it produces 2 ridges at a time, and that is a huge benefit. So after harvesting, you don't plow again, you don't harvest it, you just go straight and ridge. Now, when you produce cassava and then we are saying you harvest rapidly, it means there will be a lot of volume to also process by peeling. And cassava must be peeled as the first activity, post-harvest activity, before it can be used for any human consumption. And all the benefits you have stated. So peeling has become a problem. Now I've also tackled, and you can see how this small girl is peeling. Usually a duty for women and girls. And this is actually going against SDG 5. So there must be some intervention also. And with some of my students, we are working seriously to come out with some affordable peelers that can be used at the rural level for cassava processing. So the peelers we have about a vertical one, then we have different versions of the horizontal one. And this is what the prototypes can do. This is the So this is coming out well, and then so this is we have got this one. We are still working on it to modify it so that the losses are reduced. So this is also something we are working on. Then we are working on the cassava slicer, so that when you harvest rapidly, you peel rapidly, you slice rapidly, and then you dry rapidly, so that the same day you can get your dried cassava chips, which you can use for the high quality cassava flour. But this work is known to my Vice Chancellor, the former Minister of Agriculture, Honorable Kofi Humado. We have done a demonstration in Nigeria. FAO have got delegation from 10 other countries. Dr. Baumia, and then the immediate past Minister for Agriculture are aware of it. Kofi Humado commissioned the first generation in 2013. And but the uptake is not encouraging, and I think with this conference we can actually remove some of the impediments. In fact, when this is working, I'm suffering from piracy, infringement, and then even litigation. So this has been copied by Chinese delegation that visited my farm and then produce this, and without me acknowledging it. Even right now, there's a Chinese company in the country that bought one of my harvesters. They just copied. And these are very sad aspects. Even though even those of us from this part bad at all, we can also produce something. But I mean, so that type of infringement is limited to the developed countries. So in actual fact, cassava to move this whole crop forward, I mean, we need to work on those mechanization components, because it is a climate smart thing. We need to be multidisciplinary. And then also it is a job creation, I mean, whole venture. In fact, we need to use robotics like this Hello Tractor, and then tractors are using telemetry. So we use that one to mechanize service farmers. And then we need the collaboration of MOFA and other stakeholders to train smallholder farmers in the mechanized cassava production in the country and even other parts of the continent. So in conclusion, the third mechanical cassava harvester is the first of its kind in Ghana and Africa. Africa must adopt or must develop cassava from just being a food security crop, become an industrial and export crop. And a well-developed cassava value chain represents a multi-billion-dollar market. And the harvester we have developed will unlock the huge potential of the crop on the continent. The research team needs support to train smallholder farmers and operators to adopt mechanized production technology and methodology. Because the high-yielding elite varieties our colleagues are releasing at binary and crop research, we need to be planted in a certain format. If they are planted in the traditional haphazard, there will be no benefit. And more so because of their starch content, they cannot produce the ampesi and then the fufu for the farmers, and their interest will be low. But if you mechanize the production, you see the business advantages there, and then this is going to put whole population to benefit from this crop. I mean, at the future of mechanization, innovation, and adoption, we need to establish pilot plots, like it has been said by somebody during the opening ceremony, so that we can train, we can teach people how to adopt best practice. And then there's a need also to upscale and disseminate proven technology in the nation, maybe on the continent. We need to promote science, technology, innovation, and STEM, subject to domesticate Agenda 2063, Science and Technologies Strategy for Africa 2024, and then the Sustainable Development Goals. And sadly, which is so important, Africa must patronize made in African goods to motivate innovations, by African inventors. We have the tendency to hate ourselves, to prefer foreign goods than even local goods, and that is a disincentive to those who want to innovate. That somebody has mentioned this already. Look at the size of Africa, look at all the countries that are going to it, and look at their population. And we have a lot of land to spare. But we are always being told that we are populated and we are fighting this, and we are allowing our young ones to die in the Sahara heat. Those who can survive the heat will drown the Mediterranean, just to go for polar pastures. Just a matter of tweaking our policy, and then we can turn this whole continent into, a paradise, a heaven on earth. By acknowledgement, I want to express my gratitude to Gaik and Binari for the invitation and then this opportunity. Thank you for the audience. Prof, I'm done.
Thank you very much, Prof.
Hello, Prof. Good.
Thank you. So Prof couldn't join us because he's currently in Abidjan attending another meeting, but he's made time to be with us since morning, and he'll be online when it's time to interact. We'll be able to ask questions of his invention. But his presentation is very eye-opening. I have never even thought that it would be interesting to see how our heart rate increases when we try to uproot cassava. How many of us have tried uprooting cassava before? How was it? How was the experience? Is it something you enjoyed? Okay. We are used to it. So you see how it went up, and if you look at his graphs, very interesting how we are linking innovation, technology, and even our health. Okay, so we'll come back to him and we'll interact more with him. We have a second presentation. I told her I was going to get something about her, so I've gotten. We have Dr. Cynthia Zoria to present the second one. She obtained her BSc in biology from Universidad Nacional Agraria La Molina, okay, in Lima, Peru. After obtaining her BSc, she started her career with the International Potato Center, where she managed the molecular characterization and genetic diversity analysis of wild and cultivated potatoes accessions, as well as other Andean root and tuber crops such as oca, uloko, aracaca, and yacon for 4 years. Yacon is cassava, right? It's also a tuber, right? Yes. She obtained her PhD degree in plant breeding and plant genetics at the University of Wisconsin-Madison, where she was also a graduate research assistant in the potato breeding program, assisting in the valuation of field trials, as well as phenotypic and genotypic data analysis. Her PhD research was on understanding the genetics of potato tuber, calcium, and its implications in breeding for improved quality, where she applied QTL mapping, identification of markers associated to QTL, genetic transformation, gene expression, and field greenhouse in vitro and microscopic evaluations. Cynthia worked at the Instituto Nacional de Innovación Agraria for 4 years, where she was the director of the subdivision of genetic resources. At this position, she was also the head of the Peruvian National Gene Bank that conserves more than 250 species of plants, including medicinal plants, crops, and crop wild varieties. And they are conserved in the form of seeds, in vitro culture, and field conditions. This gene bank also conserves DNA from native and introduced animal genetic resources, as well as pathogenic and beneficial microorganisms associated to the plant genetic resources. She commanded the design of an institutional database to store information related to passports, phenotypic, and molecular data. She led a CIP and INAIA partnership to collect potato crop wild relatives for the global project towards a more complete coverage of the diversity of crop wild relatives, funded by the Crop Diversity Trust. She was a principal investigator in several pre-breeding projects to identify interesting traits that could be transferred from the gene bank to breeding programs. And she was the Peruvian National Coordinator for Plant Genetic Resources, Animal Genetic Resources, and Biodiversity for Food and Agriculture for the FAO's Commission of Genetic Resources for Food and Agriculture. She has also worked as a visiting professor at UNALM for 4 years, teaching plant genetics, plant breeding, biology, and microbiology to undergrad and graduate students. Her research work has led to the identification of several germplasm accessions with potential to be used in breeding programs. And she has peer-reviewed publications and 2 book chapters. And she has trained 4 BSc and 3 MSc students from Latin America. In November 2021, she joined the plant biology and genetics, plant breeding and genetics lab at the IAEA, and that's where she currently is. So, ladies and gentlemen, next speaker is Dr. Zoria.
Obviously, using this just kind of to this channel, I'll show you. My name is Israel Mexico.
Good morning, everyone. I'm very glad to be here to celebrate the 30 years of Benari and also the 60 years of Ghana Atomic Energy Commission. And I'm very glad also to participate in this event, the Cassava Week. Let's wait a little bit for the presentation, please. Well, I think we can get started. Just first, I want to introduce you a little bit about my institution, the IEA, the International Atomic Energy Agency. In that institution, this institution is part of the UN, and we work on peaceful uses of nuclear energy in different aspects. Some of the applications are related to science and technology, safety and security, and safeguards and verification. Plant breeding and genetics is part of the nuclear science and technology area, where we have different topics, including human health, including environment, water resources. And in this area, in nuclear science and technology, we are trying to support the Sustainable Development Goals and to contribute to 9 of them. And in plant breeding and genetics, we are contributing especially to the 2nd, the 3rd, and the 13th goal that are related to reducing hunger, improving health, and contributing to adapt to climate change. We are also contributing indirectly to Sustainable Development Goal 1, that is reducing poverty. Our center is the joint FAO-IAEA Center. This is a research institution, research center that is using nuclear techniques in food and agriculture in different topics related to plants, animal health, to soil water management, also food safety, insect and pest control. In plant breeding and genetics, our main goal is to generate new genetic diversity. That's why we use nuclear techniques to generate mutations and enhance this genetic diversity so we can select for new varieties. But we cannot generate genetic diversity and expect to have better varieties if we don't use, in addition, genomic tools, molecular markers, high throughput phenotyping, and also reduce the growth cycle and improve the seed systems. So we are working on the mutation breeding as a holistic aspect in all these other topics that complement our work of selecting new varieties. The types of funding or types of projects that we have at the IEA are 3 main types. One is the technical cooperation projects, where we provide support in the form of equipment, trainings, expert missions, fellowships. Then we have another type that is more focused on research, that are coordinated research projects. And these projects are grouping 10 to 15 different institutions working on the same problem. Then we have collaborating centers that are different institutions in a certain region, for example, in Asia, in Africa, in Latin America, We have centers in each of these regions. Now we are working on a new collaborating center with Bernari. So these collaborating centers are our partners in research and development, and also in providing some training to our counterparts from technical cooperation projects. How many of you have heard about mutation breeding? I just want to, except for Benari people, I know they know about this. Okay, yes. I think most of you have heard about mutation breeding, but for those that haven't heard about it, I will quickly explain what is mutation breeding. We won't see the video because I think it will take too much time. So what's mutation breeding? It's another approach to breeding to generate new varieties using nuclear techniques or chemical mutations to induce genetic diversity. Once we have enhanced the genetic diversity, we can start selecting using either molecular markers, high throughput phenotyping, and then after several generations of selection, we can release a variety and then start the production and dissemination of this variety. So in fact, this mutation breeding is very similar to conventional breeding. And the difference is just how we generate the genetic diversity. So we generate the genetic diversity using either chemicals or nuclear techniques. And what is the basics of the genetic diversity? New genetic diversity are the new induced mutations. These mutations can occur in any cell of the plant tissue that has been treated with a mutagen. It can occur in any gene, in any part of the genome. So it's a random process. In this case, we are relying on having a large number of cells or plants that are mutated to have some probability to find the trait of interest. So it's very important to have a large number of plants that are being treated with the mutagen to be able to find the trait that we are looking for. As well, in this case, our institution is promoting the use of nuclear energy instead of chemical, because chemical mutagens can be very toxic, and if you don't know how to dispose it well, or you don't have the facilities to dispose it well, it will be really harmful not only for the researcher but also for the environment. So that's why we are promoting more the nuclear techniques, but we also provide some training if required about chemical mutagens. Some of the nuclear techniques used for generating new genetic diversity are the gamma rays. There are different configurations of gamma ray irradiators. There are large rooms or small equipments. Our equipments are small, like what you see in the figure is like a refrigerator, and it has a small chamber inside. But I know that here the configuration is very different, right? Also, we can use X-rays or ion beams. Also, there are different configurations there. Or gamma field and greenhouses. These are very difficult to find. Only right now, only Malaysia has one active gamma field. And cosmic radiation. Cosmic radiation, we are all exposed to cosmic radiation, but in low levels because we have our atmosphere that protects us, but everything that is beyond the atmosphere is exposed to cosmic rays. And we are exploring this type of radiation now, sending seeds to the International Space Station and evaluate what are the effects. I don't know what happened. Please. Yes, so once we have exposed our material, either seeds or vegetatively propagated material, to one of these methods, either gamma rays, X-rays, or any other type of nuclear energy, we have generated the mutations. But as I said, the mutations are randomly generated in any cell or in any part of the genome. And we don't know if two cells in the same tissue have mutated in the same gene. It's most likely that they will mutate in different genes. So the next process is to make sure that the mutations that you have obtained are inherited to the next generation. How do you make sure about that? You need to make selfing in the case of seed-propagated crops. So only those characteristics that are passed to the next generation will be inherited, and you can select for those. Okay? And in the case of vegetatively propagated crops, once you generate the mutations, then you have to make subcultures to be sure, okay, to be sure that these are pure lines, let's say, of the same cell that mutated in only one kind of gene. Okay? So the first part of the process is different from the conventional breeding, but the next steps we will see that are basically the same. Okay, so after we have the mutations, we need to do a radiosensitivity testing. If we are working with a new variety that we never worked before, we need to know what is the doses of radiation that we need to apply to obtain mutant lines. So we do some calculations and determine the growth reduction 50% doses, and we can work with that. In most cases, in some cases, we need the growth reduction 30, so it depends on the cultivar. And after that, we start the selection process. First, we generate the second generation, selfing the material, and then we can start selecting. We can see a whole variety of different shapes, colors, or different traits that were not there in the original variety. So as I was saying, there is difference between what we do in seed crops and what we do in vegetatively propagated crops. Cassava is a vegetatively propagated crop, so we mostly first generate the mutations and then start subculturing to have pure lines of cells, and so then we can start working with these lines, these clones, and selecting them in the field. Mutation breeding can be useful for two things, two types of research. One is for the forward genetics, that it means we don't know which genes are involved in a certain trait, but we can see the phenotype, and we can select a mutant that has a different phenotype, an interesting phenotype, and then we can use that mutant to generate mapping population or to find what are the genes involved. That's what we normally do. Most of us are working on that kind of research. But the other type of research is when we already know what is the gene causing this trait. In the case, for example, of starch that you were discussing in the previous presentation was talking about starch. Starch is regulated by some known genes already. So we can target those and see what's the effect of the mutations in different parts of this gene. Okay? So we can use mutations for both kinds of research, but mostly we will be doing the forward genetics. And what are the opportunities in cassava? Well, you already have your in vitro methods for growing cassava and multiplying cassava. Cassava is a diploid, so it's easier to breed than other species that are polyploids, but it has some limitations because it's not easy to generate flowers and it produces very few seeds. So mutation breeding really can be an opportunity to generate these new traits, and also because there is not much diversity of cassava here in Ghana, because this is an introduced crop. So that's why mutation breeding can also produce this new genetic diversity. And well, the breeding methods that are used in cassava, the conventional methods are selecting clones and evaluating their performance in different generations, and then selecting one that is performing better than the rest. But we can use other approaches to get the most of the genetic diversity that has been generated. For example, inbreeding the mutant lines to see the recessive alleles. If you do this, the inbreeding, then you can have homozygous for the recessive alleles, and then you can see new traits. Otherwise, you will only see dominant traits. Okay, so that's something to think about. Maybe you could implement this strategy too. For that, you will have to induce flowering, increase the flowering, maybe by exposing the plants to some kinds of light or different types of temperature, growing them in the greenhouses. So that's something that can be applied, but it requires some technology to be implemented. But mutation breeding can also be used, and I think that's something that you already are doing, and it can also be really helpful for cassava. So the process is starting with the in vitro material, then induce the mutation, subculture to get rid of the chimera and have pure cell lines, and then start phenotyping and genotyping. And then if you add the inbreeding, then you can generate the homozygous for the recessive alleles and have even more traits to analyze. And then you select your best performing line or clone to go to multi-location trials and finally release a new variety. Basically, the selection process in mutation breeding is the same as the conventional breeding after you have generated your M2 population. And you can use either clonal selection or inbreeding selection. What are the new traits of interest right now in cassava? Cassava has been used as a food crop mainly, and so the yield is a very important trait. But there is also a new market about processing cassava for industrial purposes. For that, the main trait is starch, because starch is used in the industry, even in the food industry or other kinds of industry. Some products that are using starch, for example, are using for the production of different kinds of beverage. Some beverages that have a colloidal consistency. Also for the production of candy, starch is used as a gelling agent. Also for the production of chemicals for pharmacy products, starch is also used as a part of the composition of these pharmaceuticals. And also for the adhesives and glues, and is a very high quality type of glue, and also it's biodegradable. So that it has many advantages. Also in the production of paper of high quality and thick paper. The thicker the paper, it has higher quality, and it's produced adding starch. And also in textiles to make the yarn thicker or the fabrics much harder. So cassava starch has a lot of potential to be a trait of interest for breeders. So for that purpose, if you want to start breeding for processing and for starch content, you need to think about this. You have to implement your lab for biochemical and precise biochemical analysis to quantify this product, starch. And also, if you want to improve nutritional traits in addition to starch, you need to have some equipment that can help you analyze very fast but precisely the composition of minerals, proteins, vitamins, and also cyanide. That is something that can make the cassava bitter or sweet, right? So some of the equipment that is used for that purpose are the near-infrared spectroscopy and also the XRF equipments. The NIRS is used for the large molecules like proteins and oils and vitamins. And the XRF is used for iron, zinc, for example, for minerals. So if any breeding program wants to select for new varieties with improved nutritional quality and improved processing quality, they should be getting this kind of capacities in the lab. Okay. Well, for processing, the main component is starch. And starch, not only how much starch the cassava has, also what is the proportion of amylose and amylopectin, because that gives you the quality of the starch. Most processing applications require high amylopectin and amylose-free material. So if you select for a variety with high amylose and high amylopectin and very low amylose, that will be a very good variety for the industry. Okay. And we are promoting also the use of rapid generation advance using different kinds of light and different conditions of temperature, humidity, in controlled conditions. This can reduce the life cycle, and also in the case of a cassava, it can promote flowering because normally cassava doesn't flower much, and also promoting the production of seeds. So in that case, you can do the inbreeding of your cassava and obtain the recessive material, and you can have more traits to evaluate. Also, cassava marker-assisted selection is getting more and more important now that there are some markers that have been associated to certain traits that are already published and available, and that you can start using those for selection and accelerating your selection. But I would warn you that this is not something that works 100%. It's just an additional information that the breeder can have to take the decision, because these markers are closely linked to the genes causing the trait, but we don't know exactly which genes are responsible. In this case, there are markers for resistance to cassava mosaic disease but we don't know exactly which genes. So it can give you some information, but you still need to do the phenotyping. And using mutant lines, there are specific methods for finding the genes associated to a trait in a mutant line. There is this mutmap method where you cross the mutant line with the original variety and then generate the F2 generation. And with that, you can, and using, well, markers, very highly dense markers or sequencing, and then you can identify the genes causing the trait. And more recently, Cassava genomic prediction is being developed. To do genomic prediction, you need to have reference genome, highly dense markers, and some kind of model to predict. So you need phenotypic data, genotypic data in large quantities, and you need to be able to generate much faster and much cheaper the genotypic data compared to the phenotypic data. If you are in that position, then genomic prediction can be helpful in your breeding program. However, I think this is not the case for most breeding programs yet, but maybe in the future it will be possible. And there is also gene editing that is being developed in Cassava. For when you know already which genes are involved in a trait, then you can modify or mutate this specific gene to change the allele, either silencing or enhancing the expression of that gene. So gene editing is also being established in Cassava, but it's at the early stages. But here, this publication is modifying or mutating the genes that are involved in starch production. And after using gene editing, they can produce amylose-free varieties of cassava. Okay? So this is what is coming in the future. Maybe your lab also, because you have very good facilities for tissue culture lab, you can start trying these methods. Once more genes are known to control certain traits, then you can apply gene editing. And the conclusions, well, mutation breeding in general, I think it's very useful in this case, especially for vegetatively propagated crops. You just need to have a very precise method of selection and screening. And in the case of cassava, if you add inbreeding to the selection process, then you can have even more variability because you can see the recessive alleles. There are other technologies that are available, like genomic prediction and marker-assisted selection, gene editing, that you can start adding to your breeding program and be more effective and producing new varieties much faster. There are all these traits for nutritional quality and processing that are important, as you have heard from the previous presentation and the other participants that were mentioning about the needs to industrialize the production of cassava. So this is a very important trait that you can start breeding for. But you need to work together, the food sciences and the breeders to do so. Well, thank you. And this is the team that we are working at the IEA in the plant breeding and genetics program. And if you have any questions or you want more information, you can visit our webpage. Thank you very much.
Thank you very much. So we had two interesting presentations. Do you have any burning questions? I have a burning question for both of them, and I'll be undemocratic here. I'll start. So one of the things that has been bothering me for a while is the low level of protein in cassava. And I've been wondering why we are not looking at mutation breeding to help get lines that are rich in protein content. What, in your view, is the main issue that is hindering that kind of mutation breeding? And for Prof. Online, my question to him is this. I'm sure a lot of our audience today are hearing of the Tech MCH for the very first time. I have known about it. What, in your view, is hindering adoption of this life-changing technology? And why is it not a key innovation for our cassava farmers currently? So maybe we'll take Prof. Online first, and then we'll come in-house. So Prof.
Yeah, thank you. Can you hear me?
Yes, Prof, we can.
Okay, thank you. Good. Prof, that is a very interesting question. First, what in my view is hindering the adoption of this life-changing technology for cassava? There are several factors, but the key ones are this one. The way cassava is planted traditionally, until that is changed, the huge population that produce cassava will not benefit. And if these are the main targets, then they have to deliberately change from their traditional haphazard random planting to planting on ridges or rows wide apart for the tractor to pass through. That is one. And also the other thing too is that why it is not, what is hindering this is, I think the reluctance of the African to consume African innovation. You know, this is known to government. In fact, this came to light after the WAP 1. And it is known at the highest circles in government then. Kofi Humadu commissioned the first, Honorable Kofi Humadu commissioned the first generation. And then that was in 2013. And the minister, the outgoing minister for agriculture, my friend, my very good friend, Honorable also he is also aware. And why this has not, been promoted at that level. And I also want to say that, this is a disincentive for the scientific community. Our politicians are always shouting on rooftop, if we are not doing anything, scientists are not doing anything. Now, this is a demand-driven technology that is developed to help our people. And the reluctance of government, the donor community to support its widespread adoption is strange to me. So I think everybody has a role to play because we cannot be, as a continent, we cannot be importing technology from everywhere, west, south, east, and except from ourselves. So I think that is a question everybody has. But I have experienced it. I have satisfied the requirement that as a scientist, we have come out with an innovation that solves a typical African problem. This adoption, I think, government must see it to promote it Because one advantage is that this thing can even be a foreign exchange earner for the country. If government supports first of all, mass adoption and then mass production, it can be exported to other countries to earn foreign exchange. And it will serve as a motivation for our teaming graduates that we produce, who now form the Unemployed Graduate Association. So it is actually something that those in policy position must take up. But as a scientist or from the scientific community, I think this is also something that will vindicate that we are not that theoretical scientists. We can produce something that, I mean, is useful. This is not a rocket science. So I've been lamenting all along about this, but it's good you also raise it.
Okay, thank you very much, Prof.
And then the other one is a follow-up is why not a key innovation for cassava? It is, but like I said, it is related to the first issue that I've discussed. In fact, I'm not able to be with you in person here because I'm in Ivory Coast now to help the Agency for Rural Development, which is ANADE. That has sent a high-level team to my university to understand the promotion of cassava mechanically. And they have invited me to come here and then train them. In fact, I could have been in the field now, but because of this webinar or seminar, I'm actually here still. So they have seen it, and then they are seen it. So it is likely they can be a typical example of a country that can adopt it. So maybe if you get one country adopting it and using it, for me, that is successful. That is a success.
Thank you very much, Prof.
There are so many things, but I don't think it's good to say it on air.
Okay. Yes, I want the second question answered here, and then I'll come to the floor. So I'm the unofficial president of the International Fufu Eaters Association. I don't know if you've eaten fufu since you've been here, but cassava is essentially starch. And so why don't we have breeding programs that help us to have very important nutrients like proteins and others?
Okay, thank you for the question. To improve protein content, I think you need to have first your biochemical methods to measure precisely the protein content. But even if you have that, there will be a limitation because you cannot process that many samples, for example, maybe 50 samples a day, but you have to analyze 6,000 lines. So you will not have enough time or capacity maybe in the lab to process that many samples. Then it's a method that can optimize time and cost should be implemented. Otherwise, you cannot use this method routinely in your breeding program. Because I think maybe the food scientists in the room can confirm this, but I don't know how many samples you can process for total protein content using Kegdal method. I think it will not be a large number because really it requires a lot of steps, chemicals. So to be applying this in a breeding program, you really need to standardize another kind of methodology. For example, near-infrared spectroscopy, that you can use just reading. It's one reading. You don't even need to grind the material. You can read directly from the root and have an estimation of the protein content. But to use those methods, the most important thing is the calibration. So once you calibrate your model for the near-infrared spectroscopy, then you can use it routinely for the breeding, and then you can breed for improved protein content. But that's the key point, to develop the efficient, cost and time-efficient methods in the breeding program.
Okay, so
yes, we always go in for cassava roots, But we know our fellow Liberians and other nationals don't eat the roots at all. They go for the leaf. In fact, cassava leaf has got about 22% of protein, which is equivalent to that of egg. So it's just a matter of adding a lot of cassava leaves in our diets so that we can augment the lack of protein in the roots.
So I'm curious, Doc. For fufu eaters, how do we incorporate this? No, not the soup. I want the.
Hello. In fact, we know very well that fufu eaters don't eat fufu with pepper. You by all means look for Kobe and some fish in addition to cassava leaf, which is equal to the egg. So, but don't even worry. There is hope because we have yellow flesh cassava that has additional nutritional benefits. When I was in Colombia, I normally eat sanchoco. Sanchoco is cassava soup made with sweet yellow flesh cassava. And it's highly tasty and I understand highly nutritious because you can get additional benefits from the yellow flesh cassava. So cassava is also coming up with other components which are lacking initially in the white flesh cassava.
So, okay. I shouldn't worry. Okay. Yes.
Okay, thank you very much. I think it's also a follow-up of the protein. If you look at cassava, I think one of the problems is the shelf life. Actually, cassava does not, when you have it, after a short time, it is gone bad. Considering the other root crops like yams and other things where you can harvest and keep it for some time, is there any possibility to use breeding or mutation breeding to enhance the shelf life of the crop?
So that's another question for you. Yes.
Thank you, presenters. I just want to find out from Prof whether they did cost-benefit analysis of the harvester, and if it is possible, the private sector can take it up for adoption. Thank you.
So 2 questions. Let's take in-house first, and then we'll go to Prof.
Thank you. Well, post-harvest traits have not been that much explored in mutation breeding yet, but I think there is a possibility to have new induced mutations that could improve post-harvest storage, but it has not been tested.
So those who are hoping to do masters and PhDs, this could be an area to look at, using mutation breeding to extend maybe the shelf life of cassava. Yes.
Yes, it is possible to increase the shelf life. When I was with Ceballos in Colombia, one of his aims is to increase post-harvest physiological deterioration. And we managed under him to increase the storage up to 3 good months. 3 good months on the shelf. The good news is that the yellow flesh cassava has beta-carotene in it, essentially for cell integrity. So if the cells are well formed, they will not break down. It's just like you have an egg in your hand, the shells are not well formed, it easily break. The same procedure with mutation breeding, they manage to ensure that the roots are having better cell integrity. By so doing, when they kept the yellow flesh cassava and the white flesh there. Some have gone beyond 3 good months by maintaining their properties. So it is important for us to know that there is a chance to do that.
Okay, so maybe we'll come back to this discussion. He'll be part of the next session. So online, Prof, there was a question for you. Cost-benefit analysis.
Yes, thank you. Thank you, and thanks the questioner for that. Yes, I've done this several times, and well, it's unfortunate I wasn't there in person. Otherwise, I have an Excel program that I could have, presented, and then we get values from the participants exactly about the by day labor cost today. You know, from the earlier presentation, I mentioned that this thing can harvest one plant per second or less, compared to 5 to 10 minutes manually. So it means there is a very high amplification. And if one person can, it means you need about 60 to 100 people to harvest a hectare. And if you multiply that by the cost of labor now, you can see how much it will cost to harvest a hectare. Now, the harvester harvests that 1 hectare in 2 hours. Even now with the escalated cost of fuel and labor, it is still very, very economic. Additional to that, it will be in a position to make cassava available in large volumes to be processed industrially. So this is coming actually to support industrial production and processing of cassava, and the benefits are overwhelming. And, additionally, because if you harvest, you don't plow and harrow, you have actually saved fuel and time and work for plowing and harrowing. If you factor all this into the cost of ownership and the cost of operation, you see that the harvester is actually a very beneficial implement that must be used by all cassava producers on the continent.
Okay, Prof, please, I would like to find out.
Thank you. Yeah.
I'd like to find out if the harvester has been able to remove the problem of farmers waiting for first rain before harvesting. That is, can we use the harvester even in the dry season?
Yes, I think that was that is actually the biggest benefit of the harvester. The harvester is ideally developed to harvest during the peak of the dry season, when it is difficult for manual harvesting. When the soil is soft, the harvester is just like a soil engaging implement, like the plow or the harrow. You cannot, even the tractor may not get a good grip. So when the soil is very soft, you cannot use the harvester. But once you plant on ridges, the harvester can be used throughout the year, because there will be no accumulation of water on the field. And once the furrows can drain the water rapidly, the field will always be friable and trafficable for tractor. But if you plant on the flat and the place is flooded or is wet, where no tractor can even go there because of lack of trafficability, then that will not work. But the modern method we are proposing now, planting on ridges, even for smallholder farmers, will make the harvesting of the cassava all year round, especially during the dry season, when the soil moisture content is dry, it's low. And the 2 videos I played on the harvesting, they were played at a time when the moisture regime of the soil were very low. So yes, the positive contribution of this is that it can harvest rapidly during the dry season, when it takes a lot of man hours to do it manually.
All right. Thank you very much, Prof. Let's give ourselves a round of applause. So we've come to the end of the first panel. We'll have a second panel. I will be stepping aside for the next moderator to take over. And this panel will be made up of 3 eminent people. Former director, Professor Kenneth Danso, will be one of the panelists. Dr. Godwin Amenope, who we've already heard from, is going to be another member of the panel. And my own friend, Mr. George Pra from WOFA, is also going to be on the panel. So can we have the panelists come up? And I'm going to put them on the spot. Yes. So they will be looking at the cassava planting material production in Ghana. And as one key thing that was talked about early in the morning was varietal release and registration process in Ghana. Our moderator is Mr. Jeffrey Niyama from the Department of Agric, Ghana East. And so I'm going to step aside for him to take over.
Thank you very much. I hope you are all doing well. Permit me to change our surnames today, because I've changed my surname to Cassava. So I'm Jerry Cassava. So you can do same for yourself. I will not give you a name. Thank you very much. Yeah, so we want to continue with our discussion on this golden roots crop. In the morning we heard that usually the main problem with cassava has to do with the availability of the planting material. The availability of the planting material. So if you read from literature, you will see that cassava, compared to other food security crops like maize and the others, have not really received the needed attention. And it's very important that we are discussing this topic here today. And so, because we need to really understand the crop and the way it goes about helping us in our food security situation, we need to talk about the genesis of it, which is the planting materials. So we have experts here who are ready to dive us into the topic. They have 10 minutes to, each of them have 10 minutes to do that. They'll give us a brief overview, and then our questions will bring up more things that we'll need to discuss and know before we leave here. So you will agree with me that they are in the best position to help us today. So we'll look at the cassava planting material production, and to do that for us will be our own Professor Ike Danso. Without wasting much time, let's give him a hand of applause as he takes over to help us with this topic. Thank you very much.
Thank you very much, MC. And, thank you, Binari, for inviting me to be part of this panel. Yes, it is true. The major constraint to cassava production and productivity in the country is lack of planting material. And this has come about because of the dramatic increase in usage of cassava in recent years. If we are producing cassava, which feeds about 357.3 million tons a year, then we need large planting materials, huge numbers of planting materials to support this planting. And it must come in the form of plantations, cassava plantations, not at the subsistence level that we are seeing. Conventionally, cassava can be seed propagated, but conventionally we are using vegetative propagation, that is the stem cuttings. And there are so many reasons for that. Using seed propagation is very advantageous for breeding purposes. 2, it limits the transmission of viral diseases and other pathogens that affect cassava. And it also enhances transport, is easily transported. But we don't use the seed for propagation because of the fact that it behaves as a pure seed. It produces taproots. And we all know that we are not interested in the taproot because the taproot will only produce one storage root, at best 2. So farmers will lose when they use the seed. So we don't use the seed, we use the cartons. But the cartons come with a lot of challenges. The reason is that it is highly perishable. We can't store it for a long time. In fact, it is difficult to store cassava planting materials when you harvest the sticks. It's difficult to store it for about 3 months. It's very, very difficult. So that is one problem of it. 2, it has long propagation cycle. It will take at least 6 to 9 months before you get lignified materials from which you prepare your cartons. So it makes it very difficult. And the other limitation is that the cassava planting material is very bulky, very, very bulky. So it is very difficult to rope it into the formal sector for production. In fact, the reason why I think Binari was invited to the National Varietal Release Committee was that I made an argument that we have seed companies that produce seed for farmers annually. And most of the cereals we have also that for maize, rice, and other things. But we don't have it for vegetative propagated crops. Plantain is example, cassava, typical example, and bananas. We don't have it. Even yam. So it makes it very difficult for us to get clean planting materials. And currently, the demand is far outweighs the distance. Now, if you are using sticks, 2 things must be considered. 1, you must consider the quality of the planting materials from the sanitation point of view. Then you also look at the agronomic point of view. For the sanitation point of view, we are talking about the infestation from pathogens. The sticks are infected with a lot of viral diseases, and most of these are systemic. Sometimes they are very difficult to be seen or conspicuously on the plant itself. So it's very difficult to detect. And farmers unknowingly plant this, and as they do that, they keep multiplying the disease on the field. The other thing that we have to look at is also the entomologic aspect. There are a lot of pests, mites, that also destroy the donor plants. And therefore, when you prepare the sticks and you plant them, either they attack the planted material. So all these things lead to low productivity in the cassava production system. So the issue is, how do we get quality planting materials to meet the high demand for cassava? We need to do a lot of things for the sticks. First, you need to look at the age at which you can harvest the planting materials for preparation of the sticks for preparation of planting materials. Not only that, you have to look at the age. The age, to what extent has the cassava stick been lignified? Sometimes, if you look at the cassava stick, you see the green portion at the upper parts and the lignified portion below. Normally, it is the lignified portion that the farmers will go for to plant. So we have to look at that. Then you also have to look at the diameter of the stick. You have to look at the length of the stick, the number of nodes, All these things are part of the things you have to consider. Okay, all these are part of the things that you have to consider. So looking at the conventional propagation, either sexually or asexually, these are the things that you have to consider, and these are some of the challenges that we have. Now we have modern methods. The modern methods are very rapid, they are very good, but they also come with their costs, and therefore we don't tend to go for it. The first is tissue culture. We are all aware of tissue culture. In fact, using cassava tissue culture, you will be able to produce a lot of planting material within a short time because it's independent of the weather. And the planting materials are also clean, virus-free, Because if you use the meristem coupled with heat treatment, you'll be able to reduce the viral load and also eliminate the viruses completely. So the tissue culture method is good for developing planting materials for cassava. But unfortunately, it comes with cost. Setting up a tissue culture lab like that of Binari is not easy. It's very expensive. Training high power, highly skilled manpower, scientists and technicians to produce also challenging. So normally, people will not like to go for it, but it is the best way to go. And what we need to understand is that this is a place where we are failing as a nation to do. This is the place where we are failing as a nation to do. Bringing in technology to enhance our production of planting material. Because if you say that the tissue culture should be used, I've been at several meetings, we have advocated for the use of tissue culture for cassava planting materials, and it has been thrown down because they are saying that it's time wasting, it takes a long time. But it's not. If you put a lab there without giving money to do the work, that's what you expect. You are not going to get much from them. So the labs have been put in place is there, but we need input, money, funds to do the multiplication. Now, apart from tissue culture, we can also use what we call the semi-autotrophic hydroponics, developed by IITA. It combines conventional cassava propagation with tissue culture by a hemi-hydroponics that is growing in semi-liquid substratum. In tissue culture, we will use artificial medium. Artificial medium. But in that one, we use a substrate. It can be a very rich compost medium. Then when you get your plantlets coming from tissue culture, you take it out there. Then you cut them according to the notes from the plantlets you have generated. Then you grow it and cover it, and you do it outside. In a growth room. But the demands for sterility and everything is not as high as that of the tissue culture. And therefore, it's a rapid way of multiplying the cassava for farmers. And this technology is patented. So if we want to adapt it, then we have to go and pay for the patent right before we can use it. But believe you me, it's one of the fastest way of producing cassava planting materials in IIT for distribution to other places. One of the things I've also been advocating for, and it came out as an offshoot from my work I did on cassava conservation for my thesis, is synthetic cassava seed production. Synthetic cassava seed production. Now, what do I mean by synthetic cassava seed production? What I mean is that when you do the tissue culture is coming, you get the noots coming. You can multiply it. When you multiply it, then you subculture using single notes, very tiny, very, very tiny, it's not even up to, it's about 0.2 millimeters. Only the notes. You encapsulate the notes with arginate beads, but you will enrich these arginate beads with all the nutrients that the cassava needs to develop. Okay, with all the nutrients that cassava needs to develop. And because of that nutrients, the bead is serving as an endosperm to the micropropagule you are putting there. When you culture it, it comes out vigorous, multiple shoots, and you have to transfer it to the field. Now, the challenge with this, let me talk about advantage. The advantage is this, is to eradicate the transportation of bulky materials. Assuming you are planting about 10 acres of cassava, and you are going to depend on conventional breeding using sticks. Look at truck loads of cassava planting material you have to carry, the cost involved and everything. But if you develop this technology, you just reduce the cassava to just pebbles. You reduce the cassava planting materials to just pebbles, and you can pack them in sacks and go and plant them. The challenge I have was that it's very difficult to store for a long time. In the lab, under normal lab conditions, 23 degrees or 25 degrees, it was able to store for 3 months. It was able to store for 3 months. Now, after 3 months, you go and culture them in vitro you see very vigorous plantlets coming out, and you can do a lot of subculture. The challenge is transfer of this synthetic seed to the field. I tried, and the survival rate was very, very low. Probably it's because I did it in a temperate region, and there was no tropical condition for the cassava to develop. And the problem I realized was that the substrate I use, rich compost material, a lot of fungus in it, a lot of other pathogens in it. So they fed on the adenate bit, which also contains sucrose to supply to the developing shoot. So what happened? So what we have to do, which I'm thinking about, I've discussed with a few guys, that what we have to do if we want to use this technology is to include in the adenate bit some kind of a pesticide or insecticide or a fungicide. Include it as we include the other growth regulators in it. And also make sure that the soil is well treated. Then we can transfer it. This is worth investigation. After doing this, developing this in 2003 as part of a chapter in my thesis, 2 years later we went to a meeting in IEA, and we invited a Chinese guy, and he has looked at the thesis and said that they were going to produce cassava synthetic seeds in large quantities, and he was giving a talk, and I could see that everything was coming from my thesis. So after that I said, you are producing my thesis. You see, so one guy said, then you can charge him for that. But this is a technology I think we need to consider as cassava producing country to one, produce planting materials at a faster rate, to reduce the burden of transportation in planting large planting materials, acreage of planting materials for farmers. In fact, when we do this, I think we'll be able to solve the problems. So in short, this is what I have to present. If we want to increase cassava planting materials, we have to go conventionally and biotechnologically. But we need an input. And also, to address the problem, we need the formal sector to come into the cassava production system, planting material production system. We have always been talking at it at the National Variety Release Committee, but we don't have any institute or any center where we say that this is a place where you can go to collect your cassava planting materials. We don't have it in Ghana. We don't have it in most countries where cassava is consumed and produced. So we need to do that. And I'm suggesting that you are here, a MOFA rep. I think the earlier speaker said we need to have cassava schools. I've some time ago agitated for development of cassava, formation of cassava development board. We've done that for cocoa, which we don't eat. We don't eat cocoa. We've done that for cocoa. But what about cassava that we eat? So we need to do this so that they will take up this challenge of ensuring that the planting materials that are going to the farmers, we know the origin. Anytime I go to the market buying cassava or on the roadside, I have a challenge because I'm not able to come out with the variety with which I'm buying. If we have this cassava development board, they'll be able to track down and know the type of cassava they are giving to the farmer to plant. And if you are going to buy, the farmer can tell you that this is Ankara, this is Afisiafi, this is Agbelima. We've done that in yam to some extent, but cassava it is not done. And we must encourage our local private enterprises to support the cassava planting material. Thank you.
Thank you very much, Prof. We'll not waste much time. We want to continue with the next one. So if you can recall, somewhere in 2012, the then minister launched a manual that will help in the release and registering of materials, planting materials. And this is found in the Fertilizer and Pesticide Act, Act 803. Now, we want to know the process that is involved. If anybody wants to release and register a variety onto the market. And we found Mr. Pra, who is with the Crop Services Directorate, to lead us in this discussion. When he's done, we merge the two and then bring out our questions for redress by our panelists. Let's welcome Mr. Pra.
Thank you very much, and thank you for the opportunity to be part of the Cassava Week celebration. I'm looking at the 10 minutes. I can deliver the release process for everybody to understand. So this is what I'm thinking. I'll categorize my presentation into the pre-process of release activities, then the release activities and post-release activities, so that becomes very simple. In Ghana, like the moderator said, per the Act 803, we have two bodies that have been empowered to see to the release and registration of varieties. And these are the National Seed Council and the National Variety Release and Registration Committee. The National Seed Council does the approval. The National Variety Release and Registration Committee recommends the variety after doing the evaluation of a proposed variety. So when an applicant, in this case, most often it comes from institutions. So if an institution like Binari has plans to release a variety, the first action is to inform the National Seed Council, or directly can also inform the National Variety Release and Registration Committee as to their intention of releasing a crop variety. This request comes with a report on the variety that is being proposed to be released. So there's a dossier. So you bring your application. It comes alongside with the dossier on the variety or varieties that you want to release. Then the whole process starts. The Variety Release Committee would evaluate or go through the report and respond to the institution that is making the application as to when they would come to the field to do the inspection, their evaluation, and recommend or otherwise. This is for state-owned institutions or actors that have requested for the Variety Release Registration Committee to do the inspection. There are other cases where an international institution would request for their varieties to be registered in country. In such a situation, either the applicant links up with an existing national research institution to do all those trials for them, then that national institution will do the application. Or the international company writes directly to the National Seed Council or the Variety Release Committee, coming together with a dossier. The National Variety Release Committee or the Seed Council will identify a competent institution. Maybe Binari, it could be Crops Research, it could be SARI, or any institution that they deem fit can do the evaluation. Then the Variety Release Committee, following the schedule of activities by that institution, would go to the field, verify, assess and evaluate all documentations, then make the necessary recommendation. So these are pre-process activities before the variety is actually being evaluated on the field. So when it comes to the actual release process, a schedule is given by the applicant. And in this case, when the schedule is given, the applicant is supposed to give information when the Variety Release Committee can actually come to the field to inspect the field at the vegetative stage as well as the maturity stage. These are two critical stages that the Variety Release Committee will do the assessment. They come to inspect the distinctness, the uniformity, and stability of the proposed variety. And in short, we call it DUS. And DUS is normally done on station. Then we have one also called the VCU, Value for Cultivation and Use. That one is done on farm. That is where you bring in farmers to do your own evaluation. They also help with the variety selection and all that stuff. So it doesn't differ much from what is being done on station. More often, we get better results on station than on farm because it's left to the farmer. And that is what we want the farmer to know and to accept when the variety is finally released into the system. The National Variety Release and Registration Committee will only recommend a variety that is distinct. What do we mean by distinctness? It shouldn't be the same as another variety in the system or any known variety, whether outside the country. So there are a couple of questions that are being asked for the breeder to respond or the breeding team to respond. When you go through all these questions, we compare all the answers with existing varieties. If we found out that the variety, the response you've given are same as any of the existing varieties, it means you've brought somebody's variety. It should differ in at least one. If all the questionnaire that you respond to are the same, it means it's somebody's variety. If it differs in one, it means it's distinct. Apart from the distinctness, we are looking at the uniformity. How uniform the crop is on the field. It could be in the shape. It could be in the architecture. So when we take cassava, for instance, if it's high branching, when you go to the field, it should be high branching throughout. You should get like 0.1 or whatever percentage different. And if the breeder is smart, would even go ahead and take that off type. If it's a shape, the shape should be uniform throughout. We don't have some being square, others being oblong, others being round. It should be uniform. If it's a color, the color is red. It should be red. And we have, I might see this as red. Somebody might see it as pink. So we have a color chart where maybe two of the variety release committee members do not agree to a particular color. There's a chart that they can use to measure. Sometimes too, when you don't go to meet the actual crop on the field as to how the color is, but it has been harvested, they would even ask you to bring pictures. And depending on the camera you use, the picture can also be deceptive. So we use the color chart to actually check all this. Scientists, you people have made us to understand that when a variety is distinct and uniform, automatically it will be stable. So for the stability, we don't stretch the whole thing. Once it is distinct and uniform in character, the stability wouldn't be a challenge. Unless over time, you've cultivated severally and the breeder needs to do some maintenance on the variety. One of the critical things we also look at as a variety release committee is the denomination. You must get a nice name for the crop. If the denomination is not okay, it puts fear in people. It brings confusion. It will not be accepted for release. Then the institution inviting the committee would have paid all the necessary fees. Whether you engage somebody to do the trials for you, that institution would have taken the fees. The fees also to be taken by the National Variety Release and Registration Committee to do all the necessary paperwork. If it is done and your variety is also consistent with the laid down procedures, then the variety is recommended for release. Seed production and multiplication plan is another critical thing that the National Seed Council would expect to see in the report of the National Variety Release Committee. If there is no plan after release, the National Seed Council will not approve of that variety. You see, we have challenged, as Prof. Danso was saying, when the cassava variety is released, and you don't have any plan for us to know when the variety will be in the farmer's domain, then it's no. Because it will take time per our multiplication technology we have. If you take away the biotech aspect and you want to go the normal practice, at least you should be getting 1:10 or 1:15, 20 if you are doing the stem cuttings multiplication. So it will take a whole, let's say a whole year. So if you see, at least we see the plan that when the variety is released today, June, by May, June next year, the variety should be in the system for farmers to use. If that plan is not there, then it also creates the fear that the breeder has brought in for him to be recognized for promotion or any other thing. That is one of the things that the National Seed Council usually say. These people, they want promotion. That is why they are bringing their varieties for release, and that is all. There have been a number of varieties that are not being promoted as it stands now. Prof. Danso has been a member. Honorable Director is also a member. And he's always complaining that we help you release the variety, but we are not promoting it. Then the breeders intend to shoot Mufa in the leg. That Mufa, you are the extension bit of this whole process. So you need to push forward the varieties. We are in talks with those in charge, our policy makers and our ministers, to actually help support this whole process run. I'm very happy that Prof. Bobobi said that they want to industrialize cassava. We have a new minister who has tasked the commodity officers. There are 11 commodities that the minister wants to push seriously as part of the Planting for Food and Jobs Phase 2. Cassava, yam, plantain. There are 3 of the commodities out of the 11. And I happen to be part of the team that is formulating a strategic plan for these commodities. We are trying to see how best we can industrialize cassava. It's a serious proposal on the ground for the minister to consider. So Binari, it is up to us to push from that angle. The proposal has been laid. Yam, we are looking at more of the export, and plantain to export and processing. But the fundamental thing we laid before the minister was the fact that planting material is a challenge. So Binari as an institution was mentioned to help in the multiplication of planting materials. We have done what we can do. Now the ball is in the court of Binari to find ways and means to meet the minister. Because I mentioned to my director that it is no. If we do all this strategic plan without looking at our planting material base, it is very critical. So I would beg my director, Binari, your name is there, Binari, to do the first phase, at least the first phase. It's starting this year, but the multiplication exercise would actually take off next year. So the lobbying and the negotiations would have to start now, so that by next year, if things are done well, the whole process will kickstart. And it's starting now till 2027. That is the plan for the Planting for Food and Jobs Phase 2. So this is just an added stuff that I brought on board to the variety release process. The Crops Services Directorate acts as the secretariat to the National Variety Release Committee. If there are any questions, if there are any challenges regarding how to release a variety, just link up with us. If there are any sources of funding that we can also look at or link the institution to, we will do our best to do that for us to release more improved varieties that will take over the ones that we think they are being underutilized or not giving us the right yields. Thank you very much.
All right, thank you very much, Mr. Pra, for doing justice to the topic. We are very much grateful. We are jumping straight into the Q&A section, and I just want to pose 2 questions, and then the audience can also come in with some questions. The first question has to do with the bulkiness of the planting material that Prof talked about. Actually, it's something that has been a problem, a worry. I remember I had to travel to Aswansi to carry a full truck of midnights to bring it down here for us to use, and I was happy he talked about it. Now, in the interim, whilst we are looking at the synthetic cassava seed production, in the interim, what do you advise or what do you suggest that we can do whilst we are still working on this particular subject? What can we do to help with this problem of bulkiness of planting materials? Then the second question has to do with the varietal promotion, which is low. You mentioned that there are usually trials before the variety is released. We want to know, because usually when the varieties are released, you see farmers saying that, oh, this one is not good. The other variety that we've been using, the anchor that we've been using, is better than this one. Meanwhile, the trial was carried out maybe in just 1 or 2 geographical locations. So I want to know approximately how many locations are the varietal trials undertaken, so that we make the whole noise about the new variety which has been released, and everybody is happy that a new variety has come, but the farmers come back to tell you that no, this variety is not helping us. The markets don't want this variety. We'd like to know how many trials are undertaken and which locations, how many locations these trials are undertaken at. Thank you very much.
Thank you for the question. The bulkiness is a problem. You are not the only one. When I go to the village and I see my colleagues planting bulky cassava materials, go and plant this, I feel for them because it's weight. And unfortunately for the women, their children will be at their back and they are carrying the load, following their husband to go and plant. It's not the best. I think what we can do, for instance, if we have large acreage of land to plant, we need to go for truck loads. What we can do is to, one, use the conventional method to prepare the planting materials there on the field. When normally cassava, when you harvest and you store for a long time, it's dehydrates and sometimes they dry off. So what we can do is to prepare the sticks. When you prepare the sticks, then you use the pre-sprouting method. Now, the pre-sprouting method involves putting the cassava sticks with the appropriate with the appropriate bags in polyethylene bags, moistened with water. Then you seal it and make some small perforations. So you see them sprouting, developing roots, adventitious roots. In that case, you'll be able to store it for some time before you transport it. But by cutting the sticks into cartons, you are reducing the bulkiness, packaging. So you put them into polythene bags and put it in the track load before you convey it. So as soon as you get to the field, you begin the planting. Once the moisture is there, it will keep it fresh for some time before they dry. But if you are going to use the tracks, then the bulkiness will be a problem. Okay, so that's the method I think you can, in the interim, use to reduce the bulkiness, because you reduce them and get enough packaging size in the track to bring to the growing centers or planting centers. Thank you.
Thank you very much, Prof.
Thank you. To add to Prof's submission, like you mentioned, we can also use the two-node technique through the pre-sprouting. Make sure that at least you have 2 eyes or the nodes on the cassava stick, so that it is not that long stem that will be used to. At least that one can also help do more multiplication. The other issue also is to identify areas that we can do large-scale multiplication. And I think during our TIMP and the WAP program, we agreed on a production line that when a breeder releases a variety, Aswanzi, Beve, Mampon, and Winchi will be the first point of call to do the primary multiplication. Then we also identified some secondary sites where farmers were trained to do multiplication. And we could realize that during that period, when the project was running, because farmers were being paid for the multiplication, there was massive dissemination and distribution of planting materials. But usually it comes with cost. When there is no project or funding for some of these activities, then the whole line that we created gets dissolved. So it's something that we can also look at. And the plan that the new minister wants to do, he is citing enclaves, targeting off-takers or people that are into this processing. So we've targeted about 5 enclaves. The Juapon enclave, where we have Sinostone. We also have Hojo for Caltech, Ayensu for Bojuasi enclave, Amantin Atebubu for is it Amantin agro processing. Then one is it Denka at Afram Plains. So when we are able to locate production sites close to this processing facilities, transportation wouldn't also be a challenge because of the bulkiness, so that farmers will be attracted to go into production. And research or breeders will also be happy to release varieties to those who are doing the production. Now on the variety release trials, before the breeder goes into even recommending the variety to be inspected and released, he does the variety, plant variety selection. You have your terms that.
Yeah, so how many locations are necessary before you release a variety? So that's breeders' work. When you finish with that one, before you approach Agric for demonstration plus, let me take you through that. Yes, we normally start with seedling evaluation trial. During that time, you identify your mutants, and there are only 1 or 2 mutants. So the stakes are not many for you to go multi-locationals. Maybe you identify about 500 of them. But individual one is a genotype. You can't have multiple genotypes which are identical. You have to make sure that they are genotypes individually. So because you have limited planting material, you can plant the seedling, do the seedling evaluation trial for 1 year at on-station, breeder station. Then the second year, because you can multiply that seedling and have 8 or more stakes, you can evaluate maybe 500 genotypes at clonal evaluation trial for 1 year. Then it is now sufficient for you to start randomization. So you move to preliminary yield trial for 1 year under 2 reps. You must see 2 replications on the field. Then you move on to uniform yield trial, or what we call advanced yield trial at research stations. We call it on-station trials. And this one is also 1 year because you have sufficient planting material to go to research stations. So here you use randomized complete block design with 4 reps. So when you finish that, you move on to off-station trials. When he was giving us the lecture, he mentioned that you must do DUS. So it is important to do that at research station, which is on-station trial. But when you move on to off-station trial, which is farmers' field trials, you are supposed to give us VCU, value for cultivation, and so that one is also 1 year. Now, these are governed by breeders' rules. The number of reps and the designs you should have are well established. You cannot deviate from that. But as to how many locations, you must try as much as possible to cover agroecological zones you have within the country. For example, in Ghana, we have coastal savanna zone, we have transition zone, we have Guinea savanna zones, as well as forest zones. So if during your on-station trial, you can have 2 locations each, the distance between them are 180 kilometers, you are good to go. So 3 agroecological zones, 2 locations, making a total of 6 locations. And preferably at different regions. So breeders don't work alone. You must have colleagues who are breeders that you have to send the stakes to. Otherwise, you suffer. If your breeding team is large, the better. Then after this, that you have to approach Agric, MOFA, for demonstration plants, demonstration field, where you have to establish the vegetative phase and the reproductive phase. They will come in and see those stages. You do PowerPoint presentation with them. If they like it, then they recommend to a minister for varietal release. Breeders don't release variety, but it's a minister that must agree that your genotype is now qualified to be called a variety. It is then that we fall under their power. They decide to squeeze us, then we have nothing to do. If they decide to pass it, then we have power. So these are the various stages that we have to go through. It is important to do multi-locational trial because one day, if a farmer said that your variety is not doing what you claim it is doing, and he happened to cultivate it outside your trial zone that you have recommended, then it simply means you can win this case at the court that, yes, I recommended this variety to be grown in so and so agroecological zone. You have grown it outside it. That's why you are having crop failure. Thank you.
Thank you very much.
So let me add that when the variety is released, maybe the director wanted to say that it is registered in a catalog. And it details where the variety is supposed to be planted or the suitable agroecologist. It recognizes the breeder and the breeding institution. It gives a brief on the DUS and the VCU so that, like Dr. Menope is saying, if a farmer takes a variety, because it comes with a dossier, he would know that this particular variety is suitable for the agroecology that I'm going to cultivate that variety. Right. Thank you.
Thank you very much. We take questions from the audience.
Thank you. My question is to Mr. Brian. Having in mind of the new law, the Plant Protection Bill or the Act 2020, with your office and with the Act, I know that the register must be kept at the Registrar General's office. So how is your committee working with the Registrar General's office or the Intellectual Property Office with the new Act? Thank you.
Thank you. We'll take the next two and then you can address it.
Thank you very much. I think Dr. Amendope has elaborated the processes that you need to go through to get a variety out. My question is a very practical one. How much does it cost to get just one cassava variety out? And is that the reason why we are not getting improved varieties coming out on a yearly or biannual basis?
Okay, please, I have 3 questions. The first 2 goes to Prof. The last one goes to the boss from MOFA. Prof, please, you talked about synthetic seeds. Yeah, and you made mention that when you were doing the establishment, or when you were trying to establish them on the field, a lot of them died. And the reason was because probably you did it in the temperate. So you are advocating that we should think along that line. How do we then, if you are thinking along that line, how do we then get them established in our, region? That's the first one. The second one is you also made mention about age and the path that farmers pick for establishing cassava. I'd like to know how does these 2 factors, that is the age and the path that farmers pick, how does it affect or influence viral disease incidence? How does it affect or influence viral disease incidence? And then my third question for our boss from MOFA. Please, you made mention in your presentation consistently that your variety should be distinct and it's underlined. Now, as a researcher, if I go around the country and I get a very distinct variety, one that has not been, discovered, one that has not been released, can I do multi-locational trial on my own and establish a trait that it has and then come to you for release? I mean, if a landrace, okay, has a very distinct trait that has not been discovered by any scientist or any institution, and I go and pick it from maybe a very an area that has not been very remote. Yeah, it's having a very distinct trait. Can I bring it to you for release? I don't know if it's clear. Thank you.
Thank you very much. So our panelists will attempt addressing the questions.
Yeah, the first question deals with survival of the synthetic seed in a tropical environment. I think when I was talking, I made mention of the fact that I didn't get high survival rates, probably because of the temperate environment. Temperatures were between 22 to 23. If I had wanted to increase the growth room temperature to about 20, 24, 25, they said, no, they can't tolerate it because I'm not the only one who is working in the growth room. So the temperatures were quite low. But I said that what probably we have to do is not only the temperature that is a limiting factor, but the substrates, the soil in which we are going to plant. And I was using a compost, highly rich compost. And you know this compost comes with a lot of fungus and other pathogens. So they will feed on the arginate feed and therefore destroy the node. And besides, the node is very, very tiny. And you know, for vegetative propagation, you need a large part to supply nutrients to the shoot that are the sprouting shoot. So the nodes are tiny. So this, when they feed on a distance, there's not enough nutrient for the beet. So what we need to do is to include fungicides. Depending, I mean, probably we have to do a soil test in your area and look at the kind of pathogens that are there so that we can include in the beet, which will serve as endosperm to the developing node. That one will address the problem, and we need to research it. Okay, that we need to do a research into that. Now, coming to the conventional method, I talk about the age of the stake we are going to prepare your cuttings from, and also the part you are taking. Now, if you look at the cassava planting material, the top part is greenish, and it's very light, and that is the part that's also easily affected by some of these insects. So it's likely when you grow that one, the sprouting efficiency is low. The vigor is also low. And we therefore use the lignified part, the part that is quite brownish, dark. Both of them have the potential to transfer systematic viruses, systematic pathogens, because they have the conducting tissues. The xylem and the phloem are there. So as the plant is taking in this water, this pathogen might also, a nutrient, this pathogen might also be carried along with it. And they are the channels for which they transport the distance. That is the reason why if you make cuttings from a plant infested and you send it to the next field, independent of the part from which you have taken it, there will be that virus showing if it is inside. Okay, so it's not the part or the age per se, but because of the presence of the conducting tissues that are carrying the distance. That's this thing. So what we have to do then is to treat it. I know some people use heat treatment to bring down the viral load, but you can also immerse it briefly in any insecticide solution before you do the transplanting. But farmers in Ghana, we don't do that. Farmers in Ghana don't do that. So these are this thing. I hope I've answered your question. Good.
Thank you very much. If you identify a landrace and you think the landrace is unique in its characteristics or features, and you would want to work on it for a possible release, it's okay. You only have to document. At least the committee would expect 2 growing cycles. You would have done your several years of trials to actually ascertain whether it will be stable or unique in its characteristics. But at least we need 2 growing cycles data. And you are not just establishing the plot for that landrace. There should be a check. Usually, you compare with existing ones. That is why we advise that you first inform the committee as to your intention to release the variety. They might even advise you what your breeding objective is. If you're able to come up with your breeding objectives, then they will suggest similar checks for you. So right from the onset, the committee is with you and you are working alongside. So it becomes very easy for them to accept because they already understand what you are doing. How close are you working with Registrar General's Department? Act 2020 is different from Act 803. Act 803 is purposely for the commercialization. when a variety is released, how you can commercialize it. That act gives you that power. Act 803 gives you protection. Sorry, Act 2020 gives you protection. That is the one being handled by Registrar General's Department. The good aspect of it is that if we are able to well document the characteristics of your variety, Registrar General wouldn't go through the tedious exercise of allowing the breeder to go and establish the plot again, because they are looking for DUS and VCU. And usually, per the UPOF law, they usually would even dwell more on the DUS, the distinctness, uniformity, and stability of that commodity. So if they get the report from the National Variety Release Committee detailing all the requirements in respect of the DUS, then you are safe. It goes through their protocol, whether you actually deserve to get a protection for that variety. When a variety is released in a country, after 4 years, if it is not protected, it becomes a public good per Act 2020. So all the varieties we have released over the years, we are trying to see how best the new creations could get protection. So any variety that was released before 2019 does not qualify for protection. It has now become a public good. So any variety that a breeder releases or institution releases and want protection quickly would have to inform the Registrar General's Department for that protection. If you also have that intention, we could also link you up so that the whole process of having it just released could be factored into Registrar General's Department's release process for the protection. Because automatically, if you go to them for them to do the assessment for your protection, they will write back to the National Sea Council that Binari has applied for protection for Banchehine or Ampesihine. Ampesihima. Okay, Ampesihima. So the Ampesihima, if we didn't go through the DUS, it means those plots would have to be established for Registrar General to get the detailed DUS or the mandated DUS before they give protection to Ampesihima. Commercialization, Act 803, protection for Act 2020. Dr. Daniel, it will be very difficult for me to say it will cost 100 Ghana cedis to release a variety. Sometimes the location alone differs. You know why I'm saying this? I'm saying this because the pattern that we have taken, that the breeder or the institution should establish the plot for the Variety Release Committee to inspect. So you are only going to pay for the charges for their inspection and evaluation. Unlike an institution bringing it from outside the country, I would identify binary or crops research. The National Variety Release will identify binary or crops research or any accredited institution to do the evaluation. So binary will then bring the cost of evaluating that material. So that cost goes to the one who wants to do the release. So if, let's say, we even share same documentation with binary and cross research to do evaluation, the process maybe binary might take to evaluate will be different from what cross research will do. So the prices or the cost might differ. Still difficult because binary wrote a letter to the chairman of the National Variety Release Committee to do an inspection and get their variety release. The chairman instructed me to do a draft cost. in my mind, because Binari is in Accra, I was thinking the station is in Accra. I did a costing for the Accra to be the inspection to be done on station at Binari. Meanwhile, the station was in Cape Coast. So it has changed the whole cost component because committee members are coming from different angles. So let's say between 30,000 and 50,000. Yes. It shouldn't go beyond 50,000 Ghana cedis. And if you, yes, it includes the cost of evaluation, the per diem, the night allowance of the committee and all those. Again,
Chief, it goes beyond that because at the institutional level, once you have a variety you want to release, you will need to do, as Dr. Minope said, the on-station trial, the farmer field trial in multi-locations. If you add all that cost to it, it is way higher than just what it takes for the inspection to be done and then it is registered. So that's why it's difficult to really say this is how much it will cost. Because if I'm doing multi-locational trials and my institute is in Accra, if I have to do in all the 3 locations, my cost may vary from somebody at Cross Research who is centrally located and have to do in all the ecological zones. So it will be difficult for you to say one cost or an average for releasing a variety. But in all, you will need at least 500,000 Ghana cedis to go through all this process.
Just a small addition to what my director has said. You see, breeding of cassava take at least 8 years. So get it in this perspective. Breeding of cassava takes at least 8 years to 16 years. So after 8 years of research, you must pay all researchers per diem for evaluating at different regions. You must pay for biochemical analysis, molecular analysis, soil analysis. You must also pay MUFA for varietal inspection. That is what he's saying. That one alone is around 50,000, depending on the fuel price of the in Ghana. So, you know, it goes beyond the figures that they are saying because it's years of research, consistent research. But the good news is that with mutation breeding, you can even shorten the breeding period to 4 years and release your variety. So you are going to cut down. But if you are using conventional method, you are going to spend a lot of years. So it is very difficult, my friend, to give you a figure since the dollar rate fluctuates with Ghana cedis.
Okay, thank you very much.
Last one, we also know that breeders are burdened. So the committee has made things also a bit flexible, that the breeder could stagger the planting so that instead of having 2 separate inspections, which will be doubled the amount that I'm even quoting, at a go, we could inspect all these phases. Thank you.
Okay, thank you. So we'll take the last round of questions and we are off.
All right. Thank you very much. My question goes to Mr. Pra. Yeah, please, I would like to find out, can a variety be withdrawn after it has been duly registered and released? For instance, if the traits are found to be different from those reported by the breeder. Thank you.
Also to Mr. Pra and by extension, Prof. and Dr. Minope. Between cassava and yam, I want to know in this country, which of these 2 crops have the higher number of varieties? between yam and cassava. Before you even answer, my suspicion, I may be wrong anyway, but my suspicion is cassava possibly has more varieties than yam. But when you go to the market and you want to buy tubers of yam, you, even the buyer, I mean the customer, you go with a named variety. You are going to buy Pona, you are going to buy Asana or Araba or like, but this is not unlike cassava. You just go and you go buy cassava, like without any named variety. And I think this may also be contributing to why farmers don't seem to be chasing a particular variety, because they are not popular. Pona is very popular, Araba among others, I think Community and all that. When you go to your market right here at Chachu, we definitely go with. But cassava, I just go and buy cassava, and whether it's for Fufu or Mbesie, you don't have any named one. So why? If cassava has more variety than yam.
So the very last question for this session.
Okay, thank you very much. Mine is a very simple question. Now, my question to the one from Amofa, that you agree with me that before a variety is released, there are other players, just like the agronomists, the entomologists, and the food scientists, sometimes also the physiologists. Do you ensure that before these varieties are released, or before you give the certificates, these other players are well acknowledged? Thank you.
All right, so we'll turn the mic on for panelists to attempt answering the questions. I think the first one was on withdrawal of a variety. Withdrawal. So I think that goes to Mr. Pra.
Can a variety be withdrawn from the catalog? Yes. If it is to satisfy the condition of the distinctness, uniformity, and stability. After a time, if you think that the variety is broken down per the breeding objective, it's supposed to meet a particular demand or need, it's no more satisfying that it can be withdrawn from the system. There were certain scenarios where farmers came out to say that AB varieties were not meeting the demand again. So the breeder had to be called upon, and he's now doing maintenance of that variety. Again, if there is no maintenance of the variety, it's part of the conditions. Usually, once a while, the National Seed Council will go and check whether the varieties are being maintained, whether in cool store or what's the term you use on outside, SC2 or in situ? SC2. Yeah, they do all those checks. Then you misled them. You give false information. So the variety can be withdrawn from the register. We have more cassava varieties than yam varieties. Yam, for now, we have 7. 5 have been, additional 5 have been recommended for release, yet to be out. I'm sure the release letter will come soon. Cassava, we have 31. I'm happy you've brought my attention to this issue, that yam, you can go, I want Pona, I want this, but cassava, if I go and say I want ampon, you turn banchi. So I think we should have markets. Our markets should be able to recognize what it's on the ground for us, because you might even go in to buy a high starch variety. You want to pound, and you go in for this high starch variety, which is not poundable. So we need to know all these dynamics, and I'll talk to my director and also the extension director so that we do sensitization and the promotion on that. So in the dossier, it is acknowledged. Just that in publishing the variety release catalog, we are not able to put the statistician, the mathematician, and all those people into the catalog. We only look out for the lead breeder. But in the document itself, all the breeding team is acknowledged. Thank you.
I think the other gentleman asked about why is that when we go to the markets, you can easily identify that you are going to buy Puna, you are going to buy Asana, Labroko. By Cassava, you can't do that. I think it's the problem has been with nomenclature and the nature of Cassava. Cassava is at crossing, so it's always generating new genotypes, bringing a lot of varieties. So there are a lot of diversity in Cassava than in yam. So that's one of the reasons. The other reason is that we've not been able to actually follow Cassava from research institute to the markets. For instance, Amanope has released Cassava, and one of the names is Fufuhine. That Fufuhine will be planted maybe in Accra here. If you go to Bojuase, if you go to Kumasi, the farmers will give it a different name. So it's a problem with the nomenclature. And it's very difficult to follow it up. I remember when I was doing my master's like that, I went to the village and I said I was looking for Ankara cassava. Ankara is one of the earliest varieties introduced into the country. So I asked my father, at that time he was alive, I need Ankara. So we went to the farm, and unfortunately farmers will plant a lot of varieties on the same plot of land. So, and morphologically they may look similar. So when I see this Ankara, then my mind will say, no, this is not true Ankara. Then he will point to me the rare Ankara, because he had been working with it for a long time. Okay, so identification of cassava, it's very difficult. And I've always been saying that with the introduction of molecular biology, where we can use markers to mark them, I think researchers to add this so that when it gets to the end user will say that this cassava I'm selling, or the farmer will say, this cassava I'm planting is this. And if we know that you are planting a particular variety, and the women come to buy it to sell, they also say that I'm selling this brand of cassava. To confuse us, when you go to Accra market to buy cassava, they will be beating the cassava with some kind of sticks, just to make it exude the white this thing, to convince you that it's freshly harvested. It's not true. They beat to cause wounds so that the latest will come out to convince you. So these are the problems we need to address as far as cassava is concerned. But if we have a board that is managing this, the Thailand issue that the keynote speaker talked about, they give you a variety, they know the variety they are giving to you, they have it in their catalog. So if you are producing it for the starch industry, the industry knows that this man or this farmer is planting this variety of cassava. And therefore, when I buy it, I know I'm going to get this quantity of starch. In Ghana, it cannot happen. It cannot happen because they are buying from the farmers with a lot of varieties planted together. Their yields are not the same. So we expect to get the starch quantity they produce are not the same. So we expect to get this amount of starch per this tonnage you have bought. It won't happen that you are not going to get it because they are of different varieties. Thank you.
Right. Questions were well answered. Cassava is definitely having more varieties in the catalog than yam. When you go to the market, what you call Puna in Accra is actually not Puna in the farming communities, because Puna variety has tapered tail. But farmers know the differences between tapered tail and the round tail. which is Punjo, Labrador, and so forth. But because market women want markets, and they want to capitalize on the good markets of Pune, they call all kinds of varieties in the market at Pune. That is a problem when you come to cassava too. We all know that Busunsia variety simple means that this cassava variety can mature within 6 months. Usually, the Busunsia type are having red rind color and they cook very well. So farmers know very well that Busunsia is early maturity. It matures early. But do you know that the word Busunsia refers to 6 other genotypes of cassava? Depending on the locality, they are given different names. So because of that, Chinese has come out with a code. They don't want to use local names any longer. They solve this problem by giving codes to each genotype that breeders are working on. So until the genotype is released as a variety, they are giving codes so that breeders can be consistent in identifying them as genotypes, and until they are released, they are also giving them varietal release codes. So there's problem with local nomenclature.
So we are so much grateful to our panelists. You agree with me that with this limited time, we have received tons and tons of information that will help us in achieving our golden routes for the economic development. Thank you very much for being with us. I hand over to the organizers to continue, and God bless you so much.
Please, you can do it better to our panelists. Okay, so we continue tomorrow with 2 other panel discussions, and then we open binary to the whole world, because tomorrow too we'll be doing live streaming. Good. So we are going to open binary to the whole world. So tomorrow it will be on the nutritional values of cassava. So don't go home and then decide not to come back. Make it plan, purpose, and then be with us tomorrow from 9 till 12:30, where the panel discussion will be taken off. So thank you very much. Shall we? All right, so tomorrow, the venue will be at Gaik Boardroom. Tomorrow, and then Thursday, the venue will be at Gaik Boardroom. Sorry, Binary Boardroom. Sorry, over there, Binary Boardroom. And then we will add the live streaming to it. So we'll increase the number of participants online. We have shared the link, the online streaming link. So please, also share within your network for us so that we can increase participation. And then on Friday, we'll be moving to Anomaobi. The binary, we are trying our very best to let you also be a part. So we have organized a bus. But if you can help us also by taking somebody in your vehicle, we'll appreciate that so that the numbers will go higher. All right, there will be a chief. The chief of Anomaobi has been invited. The MP has been invited. Some schools have been invited. In fact, a lot of cassava farmers have been invited. When you want to see various cassava varieties on the ground, please come to Anomaobi on Friday. Thank you very much, and let's be on our feet. Father, we thank you for today. May your name be blessed. Our plan and purposes were before you. You disposed some and then you enhanced them. We thank you for all the panelists. We thank you for the knowledge that we have gained. We commit tomorrow into your hands. May you lead us. We thank you, O Lord, for in Jesus' name we pray. Amen. Give a round of applause to yourselves. Please, our dignitaries, don't go. Our dignitaries, all our panelists, the moderators, the speakers, and then the presenters, don't go. Thank you very much. So that means tomorrow.